We are live.
Hello, everybody. Assalamu alaikum wa rahmatullah. Greeting of the day. This is Majid Al-Juraisi. I am pediatric clinical pharmacy consultant, chairman of research office, and director of clinical trials. Welcome all to the COVID-19 vaccines global challenges prospects. In this session, the first session today, we will be talking about the Saudi academic efforts for COVID-19 sciences and vaccines. It's an honor for me to be chairing this session, along with 5 distinguished speakers, Dr. Naif Al-Harbi, Dr. Abdullah Al-Qaisi, Dr. Anwar Hashim, Dr. Iman Al-Mansour, and Dr. Mish'al Al-Shadhi. So it's an honor again to have them all. Without further ado, I would like to introduce the first speaker, Dr. Naif Al-Harbi. Dr. Naif Al-Harbi is the Director of Vaccine Development at King Abdullah International Medical Research Center. He earned his bachelor degree from Al-Qasim University and his master degree from UK, and he got his PhD from Oxford University. Dr. Naif Al-Harbi is chairman and members of many committees, and one of the distinguished committees that he is participating in the pandemic preparedness leading team in the HWG at Saudi G20 Summit. He's a consultant to a biopharmaceutical company. He has published many papers and book chapter. He presented more than 20 presentations in international conferences. Dr. Al-Harbi is a principal investigator and co-investigators on various research grants on vaccines, viruses, and immunology. I would like to invite Dr. Naif Al-Harbi to give his first speech. It is about the Kmark Oxford MERS vaccine development, setting the stage for COVID-19 vaccine development. Dr. Naif, floor is yours.
Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. Thank you so much, Dr. Majid Al-Juraisi. Thank you for my colleague in the organizing committee for giving me this chance. Please let me know if the sound or the video is not clear. So I'm talking today about the ChAdOx MERS vaccine that we are very proudly working on with the University of Oxford, and the journey from lab bench to camel and human clinical trials. So we all remember MERS or Middle East Respiratory Syndrome coronavirus hit the world in 2012, and this is the epi curve where we still see this virus ongoing. Recently, we published a paper seeing this virus in camels in slaughterhouses in Riyadh, with a percentage of reaching 38% of the camels are infected. So it's still an ongoing issue, and we hope not, but it could be. It would hit or cause an outbreak in human at any point. And there was a focus on international efforts since 2012 to develop a vaccine, and there was an emphasis into developing vaccines. This comes from international committees and bodies. So in Riyadh 2015, Geneva 2017, and Seoul 2018, where the world gathered to put the plan and the blueprint of the WHO. The R&D blueprint was developed. Also, CEPI came into existence around that time, and CEPI put MERS vaccine as one of its top priority. So in Oxford, in Kmark and Oxford, we developed this vaccine in mice and moved it into human and camels. In mice, the idea of this vaccine is that you give the vaccine to camels to prevent infection into human, but also you give the same vaccine to human to prevent further infection and outbreaks. And there were three main leading candidates. One of them is ChAdOx-MERS. ChAdOx-MERS was developed by taking the spike protein from the MERS virus into a chimpanzee adenovirus called ChAdOx, generating ChAdOx-MERS, which is a viral vector vaccine. And we know that viral vector vaccines have an advantage over many other platforms by being able to infect the cells and induce immune response, cellular and antibody as well. So in mice, this is the data we published with Oxford, and what we showed here is like ChAdOx-MERS with TPA, or the protein secretion leading sequence, could give you more, or at least in this case, gave more immune response in terms of antibodies as well as neutralizing antibodies. So this molecular, I wouldn't call it molecular adjuvant, but molecular enhancement helped us to find and tune, optimize the ChAdOx platform. In mice, efficacy data done in the NIH and Oxford, it was showing that 100% efficacy or protection in these transgenic mice. So we moved to another collaboration with the Ministry of Agriculture and Oxford and KMark, as well as a number of other collaboration in the world, as well as internally, King Abdulaziz University, into developing this vaccine, or at least assessing this vaccine in camels. This project took us almost 4 years from conceiving the idea and setting up the stage, building up the lab, building up the research farm, camel research farm, until we published our paper report last year, 2019. So this is part of photos of our setup. It's a very isolated camel research farm where we gathered the team to test, to vaccinate camels, and different camels, and I'm going to show the data now. And the idea is like 20 camels, some of them naive in green color, some of them red, seropositive in the red color, challenged after 3 months with those black camels representing infectious camels to assess efficacy. We can look at graph C, where we see increase in the antibody response in the seropositive camel vaccinated with the vaccine, with the ChAdOx-MERS. And this immunogenicity up to day 84, after day 84, it's the challenge day, so you expect every camel to show a response as a result of the natural infection. So this immunogenicity contributed to protection. We assist protection in 2 ways. On the left side, we see the nasal secretion, and on the right side, we see the PCR viral load in the nasal cavity. And over time, we see a reduction, significant reduction in the viral RNA load in these camels. We published this paper, and we found the conclusion is like 90% of camels in Saudi Arabia are already seropositive, having pre-existing antibodies. So it may be, so these pre-existing antibodies do not confer protection from reinfection. So it might be a good strategy to vaccinate seropositive camels to enhance or to increase the coverage of this immune response in camels in Saudi Arabia. Single dose of MERS was safe, induced higher antibody levels, protect camels in challenge model, and the challenge model was a very strong challenge model. So for sure, we assess the efficacy by mixing 10 camels, but still, over the course of the study, the number of infected camels reached 23 camels in a very small vicinity. We moved into clinical trial, collaboration with Oxford, and support and regulatory supervision from Saudi FDA. So the plan was to do phase 1 in the UK, and that was completed and published, and to do another phase 1B in an endemic country, which is Saudi Arabia here. The trial started last year, but it took us like 3 to 4 years to set up the trial, going through the regulatory pathway. And this, since it was the first phase 1 clinical trial, it was a new experience for everybody, including the regulatory or the regulators. So this is part of what we have been through to set up the clinical trial, all of the GMP certification, GLP, the clinical animal model, and so on. We also built the team and facility. We have the phase 1 clinical trial clinical unit or ward where we can further embark on other clinical phase 1 trials. We started vaccinating in December 2019, just before COVID, and we completed the recruitment in July. We're just waiting for some follow-ups to conclude all the data. So what I have shown is a very quick snapshot of effort being done in 2 different countries by Oxford and Kmark to set up the stage. Well, we did not know about the pandemic prior to it happening, but what happened is like being ready, and that's what SAPI and other organizations are set up for, as to be more prepared. I think ChAdOx vector benefited from having this TPA enhancing leader sequence. So when the COVID came, the ChAdOx COVID, we heard yesterday from Sarah Gilbert and other presentation that ChAdOx COVID vaccine is having this enhancing sequence. We also optimized the dose. So in ChAdOx MERS, we used 3 different doses, while in ChAdOx COVID, we already know which the maximum dose that's still tolerable and did not induce severe adverse event. So I think this helped a lot in setting the stage into developing a vaccine for COVID. In addition, here in Saudi Arabia now we have our phase 1 clinical trial approved, as well as the site in Kmark, the clinical ward. Also establishing clinical and laboratory team in Kmark for vaccine clinical trials. And at Oxford, I'm sure there has been much of better prepared teams and infrastructure when COVID hits because of the history of working on ChAdOx, MERS, and other emerging pathogen vaccines. I would leave it at that. I would thank so many people here without mentioning their names who contributed to our CAMEL studies, as well as those people who contributed to our clinical trial study. And these are our partners. Thank you so much.
Thank you very much, Dr. Naif. And moving to the second speaker. Our second speaker is Dr. Abdullah Al-Qaisi. Dr. Abdullah Al-Qaisi is an assistant professor in virology and vaccinology, University of Jazan. He's visiting also at University of Texas medical branch. Dr. Al-Qaisi is going to talk about the rapid development of SARS-CoV-2 serological assays for vaccines evaluation and seroprevalence study. Dr. Al-Qaisi, the floor is yours. Dr. Al-Qaisi, can you hear us?
Yes, can you hear me now?
Yes.
Okay, sorry, I was muting actually. Thank you, Dr. Majid. I would like first to thank the organizer for this really great and timely meeting about COVID vaccine, and especially for this really great track about highlighting the Saudi academic efforts in response to COVID pandemic. And that's what I'm going to talk to you about. I'm going to highlight some of the national effort. Really big team have been doing amazing work. And the title of my talk is going to be about rapid development of SARS-CoV-2 serological assays for vaccine evaluation and also for seroprevalence studies. So I'm going to talk about 3 things. First is in-house ELISA that's been developed and validated in our lab, and then another pseudovirus SARS-CoV-2 for neutralizing antibody evaluation, And I will end up with a study we just did to understand the humoral immune response in COVID patient in multiple cities in Saudi Arabia. So this is the theme is this work is part of a huge national collaboration in response to COVID and compromise of these universities. So why we need to establish in-house ELISA? As we know, when the pandemic hit, there was nothing about SARS-2. Also, they were really the supply chain impacted heavily. So we need to have hours and ensure the constant supply of the test, which allow us for rapid evaluation of our candidate vaccines, monitor and understand the antibody response in COVID patient, and also for seroprevalence studies. So as we know that SARS-CoV-2 has 4 structural protein. One of them is this spike protein, which is composed of the S1 and S2. S1 has the receptor binding domain, which is the major target of the neutralizing antibody. The other important protein is this nucleocapsid. So nucleocapsid is the most abundantly expressed protein. It's highly immunogenic. It's target for most of non-neutralizing antibody. It's actually easily expressed in bacterial system. That's why we decided to target these 2 proteins. So this is our timeline. So when the SARS was discovered, the sequence was also announced in January 11th. We designed and ordered the plasmid. By March 31st, we got them, and we received the COVID patient and started our validation studies. So and now we are at this stage where we're doing seroprevalence, and we are also doing immunogenicity for candidate vaccine, as well as screening of entry inhibitor using these assays that we have developed. So once we purify the protein S1 and N of SARS-2 in-house, we tested their capacity to bind to serum samples from COVID patient. As you can see in the left panel, that's S1 purified S1 was bind to the COVID positive sera, while COVID seronegative was not binding, indicating that the S1 was specific and successfully purified. And same thing with the N protein. Further, we did the optimization and determined the cutoff, so we can use this assays in large scale. As you can see, we determined the cutoff of the S1 IgG, S1 IgM, N IgG, and N IgM. After that, we tested their cross-reactivity with other COVIDs, and as you can see, none of the either S1 or N bind to any of the common coronaviruses, not either for MERS, indicating this S1 and N is highly specific and has no cross-reactivity with SARS, with other coronaviruses. Just want to move on quickly because I have a lot to cover. So this is again the specificity and sensitivity. And what we found is that for serum collected from week 3 to 4, we can reach up to 100% sensitivity of our assays, especially for IgG. But IgM, as we know, is start to decline by week 3 and week 4, which result in a reduction in sensitivity of our assays. So that was about the ELISAs developed, and then about the same time, we started working on an assay to establish an assay to evaluate neutralization antibody for SARS-CoV-2, and we use the recombinant non-replicating Thi-skewed to express the SARS-CoV-2 S gene, and that Thi-skewed has luciferase expressed. This system is very simple and can be used in BCL3. There is no need for BCL2. There is no need for BCL3, which is really limited in the country. Recently, we have a new version of this assay in which we increase the yield of the virus rescued for more than 1 log. So this has been published. I don't want to go to the details, but you can read our protocol. It's very detailed, step-by-step protocol in how to establish this assay by yourself and then use it for evaluation of neutralizing antibodies. So just to summarize what I have just mentioned, so we developed and validated S1 and N protein-based ELISAs for detection of SARS antibody. These assays have high sensitivity and specificity and did not show any cross-reactivity with other CoVs, currently produced in-house in large quantities, and we are happy to share reagent and protocols with others. We also established a robust neutralization assay based on SARS-CoV-2 S pairing VSV pseudovirus. Currently, both these assays are used to evaluate several SARS-CoV vaccine candidates, as well as seroprevalence study in multiple states in the kingdom. And my colleague, Dr. Hashim, is going to talk to you about some of the vaccines that we are now analyzing using these assays. Lastly, I'm going to talk about a recent study we just finished and just submitted, and it's under review in one of the journals. It's about the kinetic of humoral immune response in COVID patients with different disease severity. So as we know, COVID is still spreading, and there is urgent need to conduct in-depth analysis of humoral immune response in COVID patients. So in this study, we aim to understand the kinetic of IgM, IgG against the S1 and M protein, as well as understanding the neutralizing antibody in COVID patients with different disease presentation, for example, mild, moderate, severe, or different outcomes, those who survive the infection compared to those who succumb to the infection, using our assays. So what we did is we collected a total of 240 serial serum samples collected from day 4 all the way to day 70 from different patients, as I mentioned, and we did the ELISAs for S1, N, as well as neutralizing antibody. So I'm just going to highlight the most important data. As you can see here, we analyzed the serum sample up to day 70, and across all patients, all our cohort, despite the disease severity, we found in most patients induced sustained antibody response. There is a peak at day 2040, but after that, a small decline, but sustained until day 70. And that's with IgG against S1 and N. Of course, IgM start the decline, which is normal after day 40. Small detection limit by day 70. Same thing with neutralizing antibody, as you can see in the top. They reach the peak by day 40, and then they start the decline, but maintaining really high level up to day 70. And you can see in the right panel, this is selective patient in which we follow them throughout day 70, and we found basically almost all patients have sustained antibody until day 70. There is one from the mild, which is shown in green, that start to antibody response, especially neutralizing antibody, decline completely, and one of the severe, but that could be an outlier. And the second important data we found is that so the level of neutralizing antibody was correlated with the level of S1 response, but not the anti-N antibody. Lastly, we found that the disease severity actually impacts the antibody response against SARS-CoV-2. As you can see here, we found the majority of severe cases, which is shown in red, have the higher response, have the higher antibody response across all the antibody we tested, either IgM against N or IgG or the neutralizing antibody response. Same thing we observed with the ICU versus non-ICU, or those who died versus those who survived. Again, this data indicate that the disease severity actually correlated with the highest induction of antibody response. The last data I want to share with you today is that we found that early high level of anti-N response, as you can see here, could be associated with the disease severity. As you can see in both severe ICU as well as those who died, we see significant induction of anti-N antibody, especially in the first 15 days. That's the acute phase, which indicate that antibody response could be predictive of prognosis of the disease. So just to summarize, our data indicate that COVID patient can generate high level of antibody response that might last for a long period, in our case for 70 days, and we're still following some of these patients. Also, we show that severe infection are associated with overall stronger anti-S1 and N, as well as neutralizing antibody compared to those with less severe symptoms. And most importantly, we show that significantly higher level of anti-N actually was observed during the acute phase of the disease in severely ill patients and those who enter ICU or succumb to infection compared to the rest of the disease categories. And with that, I just want to thank everyone contributed. It's a huge effort from King Abdulaziz University, Design University, Taiba University, KAUS, Tabuk University, and KMARC, and also Ministry of Health, multiple hospitals actually contributed with sharing these samples with us. And that's all. Thank you so much, and thank you for your attention.
Thank you very much, Dr. Al-Qaisi, for such an outstanding work. Now, moving from the general principles about vaccines into more specific topics, and let's start with our speaker, Dr. Anwar Hashim. He's the Deputy Director and Associate Professor in Immunology, King Fahd Medical Research Center at King Abdulaziz University. Dr. Hashim is going to talk about rapid development of several SARS-CoV-2 vaccines based on recombinant plasmid DNA and adenovirus vector serotype 5. Dr. Anwar, the floor is yours.
Assalamu alaikum. Thank you, Dr. Majid, for the introduction, and I would like to thank the organizing committee for inviting me to present some of the work that is being done at King Abdulaziz University in collaboration with many institutes and universities across the country and even internationally. What I'm going to talk about today is what we're trying to do in our research group to build capacity within the university and within the country to develop vaccines and develop every needed reagents or for such purpose. And I think the part that presented by Dr. Abdullah Al-Qaisi is just an introduction and part of what we have been doing since the pandemic. Basically, we had already established platforms in our research in our lab, and that we utilized right away after the pandemic. We looked at the published sequences, and actually we looked for developed consensus codon optimized full spike protein from SARS-CoV-2, in which we also played with the secretion signals to enhance production and eventually synthesize that synthetic sequence, cloned it in our acceptable vector for clinical use, and then we confirmed expression and the cloning and by sequencing and restriction digestion. Eventually, of course, we confirmed that by expression of the protein in cell lines and by immunofluorescence, as you all see in here, compared to the control. And of course, this work led by Khalid Al-Haybi and Sousan Al-Amri, PhD student in the lab, started right away looking at the immunization in animals, and they started working on 2 animal models for immunogenicity, including bulb C mice, the white here, and the black 6 mice at the bottom, and immunized mice with 3 doses 2 weeks apart, and started looking at the immune response. And as you see in both cases, the vaccines induced antibody response as early as week 4, which is 2 weeks post second dose, and the immune response enhanced significantly after a 3rd dose, and as we see here on week 8. then they started looking at the different isotypes of the antibody response in terms of IgG1, IgG2A, IgG2B, to actually determine the type of the immune response, if it's Th1, Th2, and that's based on what we've been observing and we have developed before in terms of vaccine for MERS coronavirus. And as expected, these vaccines induced Th1 skewed antibody response in both my animal models, although it was more pronounced in black 6 mice as expected because of their genetic background. Then we looked at neutralizing activity, and here is shown in both animal models, and here is shown individual neutralizing antibodies using for each mouse in the test group or the control group. And this is utilized the pseudovirus assay that was developed by a postdoc in the lab, Sarah Al-Mahboub, and presented to you by Abdullah Gaisi in the previous presentation. And importantly, the vaccine induced significantly high levels of neutralizing antibodies with a mean of around 10 to the 3 in both animal models, suggesting that these vaccines could be highly immunogenic and induce protective immunity. What was interesting is that when we tried to compare this vaccine using different immunization routes in mice, and this is in bulb C, we didn't observe any response using intradermal or subcutaneous immunization. And the second important point is that the antibody response was long-lasting and observed up to week 17, which is around 120 days post-immunization. And I think now we have data from 160 days post-immunization. So the vaccines is inducing sustained immune response, but unfortunately it failed to induce response via subcu or intradermal immunization. So at the same time we were doing this work, we started to collaborate with a pharma company in the US called Pharmajet, which actually they developed a needle-free system to enhance immunogenicity and to minimize the number of doses, and in which they had Tropus and Stratus systems. They're mainly for human use or large animal testing. So in our collaboration, we developed a prototype for small animal model testing, and in which we used Tropus as one system just for both intradermal and intramuscular injection. Using this system, we tried to work on our vaccines, and we tried immunizing the mice intramuscularly or intradermally. And interestingly, intradermal vaccine this time gave very strong immune response even after 2 doses here. Here is week 4 is only after 2 doses, and it was in a dose response pattern, indicating that the intradermal, the needle-free system could actually improve the immune response in using DNA vaccine. And here is also similar data using needle-free versus needle injection using intramuscular immunization with different doses, 25 microgram, 50 microgram, and 100 microgram. We saw a trend of dose-dependent immune response and enhancement of immune response using the needle-free system compared to regular needle injection. Apart from using DNA vaccine, and this is ongoing study now, and we're also continuing the work, and I will let you know exactly what we're doing at the end of this presentation. But I would like here to move to the second platform that we developed over the years in our lab, and which is a adenovirus viral vector based vaccines, in which we're using adenovirus serotype 5 to express highly immunogenic and targeted vaccine against different pathogens. We use this platform against flu, And as you see here, we used very weakly immunogenic protein from influenza, which is the nucleoprotein. But using our platform, we were able to actually enhance the protection and induce heteroceptive immunity in animal models, in mice, against different viruses. Again, we utilized it using the HA2 subunit from the influenza hemagglutinin, and we enhanced the immune response and induced heteroceptive protection in mice against H1N1, H3N2, H9N2. The platform is basically using any antigen and making it secreted and targeted to specific cells in the immune systems that we all know as antigen-presenting cells, including macrophages and dendritic cells, which actually we found it's not only targeting and focusing of the immune response, but also enhances the immune response quality and quantity. And recently, in last year, we published this work in collaboration with the University of Texas Medical Branch in Houston, in Galveston, where actually we used this platform to develop a vaccine against MERS-CoV, and we observed similar pattern, and most importantly, we observed less severe disease in the targeted vaccine compared to untargeted vaccine, although both vaccines, S1 and S1 CD40 ligand, conferred 100% protection. So right away, sure. Right away, we developed a platform, used this platform to develop a vaccine for COVID using S1 from the spike protein. And we're currently comparing this immunogenicity of these vaccines in mice. It's ongoing study led by one of the PhD students, Najwa Al-Jahani. And immunogenicity is being tested in baboons in collaboration with King Faisal Specialist Hospital in Riyadh. And we're also testing protective efficacy in permissive mouse model, which is Syrian hamsters in collaboration with Health Canada and National Research Center in Ottawa. In conclusion, I think we developed a naked DNA vaccine that is immunogenic and elicit Th1 skewed immune response in different mouse models. We enhanced the immunogenicity of these vaccines using the needle-free system to induce robust dose-dependent humoral response via different routes. Previous work established adenovirus platform represented a promising approach to quickly develop safe and effective vaccines against emerging viruses. And finally, now we're testing cellular immune responses in terms of CD4 and CD8 T cells of the naked DNA vaccine, and also evaluating the S1 DNA vaccine, which is another version of the vaccine that we're developing in addition to different encapsulation using nanoparticles and liposomes. And finally, the adenovirus platform is being tested currently in our lab for immunogenicity and in collaboration with different labs for protective efficacy. Finally, I would like to thank the great group who actually helped with all this work. Everybody played a great deal in delivering this work. And also, I would like to thank collaborators from Health Canada, from University of Texas, NRC, SaudiVax, Pharmajet, King Abdulaziz University, King Abdullah University, Tabuk University, Taiba, Ministry of Health, Jazan, King Faisal, and KMARC, of course, and also the generous funding from King Abdulaziz University, Ministry of Education, and CAXT. Thank you very much.
Thank you very much, Dr. Anwar. Now, as a continuation of the previous talk, we will be having the next speaker is Dr. Iman Al-Mansour, who is Assistant Professor at the Department of Epidemic Disease Research Unit at Imam Abdulrahman bin Faisal University in the eastern region of Saudi Arabia. Dr. Iman Al-Mansour is going to talk about the newly developed plasmid DNA vaccine for the COVID-19. Dr. Iman, the floor is yours.
Bismillah ar-Rahman ar-Rahim. Thank you, Dr. Majid, for the introduction. I would like to thank the organizing committee for this opportunity to present our work and highlighting the Saudi efforts in COVID vaccine development. So my talk for today will focus on our preclinical study for plasmid DNA vaccine against SARS-CoV-2. So one of the key advantages that we used here is that we applied the plasmid DNA vaccine technology in the development of our COVID-19 vaccine. And DNA vaccines offer several advantages over conventional vaccines, and these include its selectivity to contain a single gene of interest, and also the manufacturing of plasmid DNA is rapid, generic, and can be produced in a larger scale and in a short time. Also, plasmid DNA is known to elicit both humoral and cellular immune responses, and also its high thermal stability, which makes it of a particular interest during vaccine shipment and storage. The vaccine constructs we used in our plasmid DNA was based on we have produced two different forms of the spike, one that is full-length, codon optimized, and the other one encoding only the S1 subunit. And the gene selected for our study was because it is known that the spike glycoprotein is a major antigen.
Can you enlarge, put full screen for your presentation? See at the bottom, the small, yes, go down.
Stop sharing.
No, not stop sharing. Yes, go down a little bit.
I put it now as a full screen.
Sure.
Okay. So we have already, to improve the translation, the transcription and translation efficacy of the our constructs, we optimize constructs by using a codon preference, using the mammalian codon preference for Homo sapiens. This also includes further modifications like the GC content, the mRNA stability, as well as the encrypting, splicing, and the premature polyadenylation, and RNA instability motifs, and several other modifications. Besides, other fragments were included to enhance the translation efficacy of our vaccine. So what we have done here is that we immunized the mice either with 3 doses or 4 doses with vaccine.
For interruption, but your slide is not moving, so we just see the background. Can you make it live? Show. If you can go down to the bottom of the screen, you see the.
So I'll go back.
Yes.
Can you see it now?
Yes, now we can see it.
All right. So we have done either 3 doses or 4 doses immunization, and using 100 microgram of plasmid DNA, either the S full length or the S1. The mice received the vaccine prior in the first week, and then they were bled 2 weeks after each immunization. The antibody responses was collected for blood and serum evaluation 2 weeks after the last dose from either the 3rd or 4th doses. So here we have roughly showing the ELISA results for our codon optimized S full length after 3 immunization, and with 4 immunization. We could see that 3 immunizations were sufficient to elicit potent immune responses using the full length S gene. While in S1, the immune response was in a moderate level. The 4 doses enhanced somewhat the binding antibody responses following 4 immunization. With the 3rd vaccine group, we can see there's just slight or moderate immune responses elicited after either 3 or 4 vaccinations. For the neutralization, we have done the surrogate virus neutralization test as a quick evaluation for the antibody titers that can show the levels of functional antibodies that binds to the ACE2 receptors. Only the full-length vaccine showed potent immune responses either after 3 or 4 doses, while in other constructs, the levels of neutralizing antibodies were not that high. So to conclude in this part, we all know there is a global demand for the development and deployment of a prophylactic vaccine, and DNA possesses numerous features that are ideal in pandemic situation. In our study, we showed that only 3 doses were sufficient to elicit a high level of binding and neutralizing antibody responses for SFL, while in S1, 4 doses were required to elicit moderate levels of immune responses. In addition to our work in vaccine, we have also established a virus bioinformatic resource to help us to monitor the genetic and any possible antigenic variation, which makes it of a particular importance during if there is any immune evasion and for determining the vaccine efficacy over time. So we have recently launched a bioinformatic database called humanviruses.net. This includes a database for SARS-CoV-2, SARS, and MERS-CoV-2. In that database, the user can search for a particular gene or protein of interest at any specific host within a specific country and the date. This is the user-friendly interface for our database that is recently launched, and the user interface is also powered with a in-house standalone bioinformatic analysis tools like the cluster and also the geographic spread to see the geographic trend of a particular mutations. In addition, we have built also a standalone blast search in which you can insert any new sequence and look for any similarities with the built-in database we have. Since determining the antigenic epitopes is crucial for finding any immune escape, we have also put a database for all of these antigenic epitopes, which catalogs the T cell and B cell antibody that are predefined in previous publications. This also includes linear and discontinuous epitopes. This database was done in a continuation for our work for molecular and immune surveillance for viruses that causes also epidemics. That has been done previously with MMR and also with ERM database, which helped us to design an improved vaccine. At the end, I would like to thank all the contributors. I would like to thank the funders by the Ministry of Education and IAU who supported the vaccine development. And also would like to thank the funders in CAGS for supporting the establishment of human coronaviruses databases, as well as our growing work in epitope mapping. And with this slide, I would like to stop here. Thank you.
Thank you very much, Dr. Iman, for keeping us on time. Now, the last presentation we have is for Dr. Mish'al Al-Shadhi. Mish'al Al-Shadhi is the assistant professor in King Saud College of Pharmacy, King Saud University. He served also as a consultant for regulatory authorities. Dr. Mish'al Al-Shadhi is going to tell us about moving from DNA to RNA. So development of the COVID-19 vaccine using the messenger RNA platform. Dr. Mish'al, the floor is yours. Can you enlarge your screen, Dr. Mish'al?
Is it okay, Dr. Majid, now?
Yes.
Okay. First of all, thank you, Dr. for inviting me today. Big thanks for Ministry of National Guard, King Abdullah International Medical Research Center, KMAR, for this such a global great annual meeting. Today, inshallah, I will be talking about our effort at King Saud University College of Pharmacy in collaboration with King Abdullah International Medical Research Center, KMAR, trying to development of mRNA-based vaccine candidate against COVID-19. So in our laboratory, we are working with two platforms for vaccine development. We are working with protein-based vaccine, and we are working with mRNA-based vaccine. For protein-based vaccines, we are collaborating with the Center for Vaccine Development at Texas Children's Hospital and Baylor College of Medicine, in which we are working with RB domain of the spike protein of the MERS-CoV viruses. Also for mRNA, we are working targeting either the full spike protein or the receptor binding domain for the spike protein against either MERS or COVID-19. Today we'll be focused more about COVID-19, inshallah. I will skip this slide since a lot of people already talk about the interaction between the virus and the human cells. Let me first talk about different achievement or advancement that make our work with the mRNA possible. So there is certain things happen in the field, including development of enzymatic codes that enable us to cap the 5' and polyadenated the 3', which enhance the stability of the mRNA. Also, development of codon optimization for mammalian expression facilitate the development of the mRNA targeting different viruses. More recently, development of different modified nucleotide enable us to minimize the innate immune system response and increase the translation yield of the protein. Finally, improvement of different delivery mechanism or systems that can purpose either protect the RNA from degradation and also increase the cellular uptake of the mRNA and make them inside the plasmid and ready to be translated to a protein. This is just a table from our recent publication about mRNA platform, compare different platforms. So there is several advantage of using mRNA. One of these advantage, including that mRNA by itself is very fast in the development and production, especially in this situation of the COVID-19, making working with them, and if you adopt this technology in your laboratory, you can extend it to other infectious disease. In addition, a multivalent nature of the mRNA, in which we can combine different mRNA strand together, targeting different infectious disease, and we see different people talking about combine different mRNA from different coronaviruses, SARS-1, SARS-2, or even MERS-CoV. mRNA by themselves, they induce really strong cellular response that can enable them to be used as a vaccine. Stability is it was an issue. mRNA used to be fragile molecules or fragile genetic materials that a lot of people they avoid using mRNA for as a medicine. However, certain advancement in the field, specifically engineer certain genetic element in the mRNA construct themselves, lead to increase the stability of the mRNA. So now we can have mRNA that's stable at minus 80 degrees Celsius for range from month even 2 years. And yesterday we heard from some of our speakers that there is an effort to make the storing the mRNA at 4 degrees even possible for several weeks, which can really save a lot of problems. Regarding the safety profile, mRNA considered to be safe because of their transient character of mRNA. They are disappear even inside the cell. They are less stable than the DNA, and also the low doses that can be given during the vaccination. And from the recent clinical studies from different groups and companies, there is no issues or significant side effect or safety issues with the mRNA. So our platform is dependent on, we start first with the cloning of our target mRNA, and here I'm talking about either the full spike protein or the receptor binding domain from the spike protein into a plasmid, and then transfer this plasmid to E. coli cells, and then we amplify our plasmid DNA. This plasmid DNA purified, and then in step 2, we linearize it and use it as a template for RNA transcription. Furthermore, in step 3, we are capped our mRNA and add the poly A tail, and that lead to increase the stability of the mRNA. Finally, we usually purified our mRNA to get rid of any contaminant, including double-stranded RNA or some amount of DNA. So for the mRNA, we are working two different mRNA, the conventional mRNA, in which we have certain features. So we have our gene of interest sandwiched between different genetic elements. So on the 5' we have the cap, which is increase the stability. In the 3' we have the poly A tails. And we play with the untranslated region in the 5' and 3' and select this region from mostly stable mRNA. And that will lead to increase the stability of our mRNA and leading to increase the translation intracellular. The other platform we are using is self-amplifying mRNA, in which it is similar to the previous one. However, directly upstream of our gene of interest, there is non-structural gene for self-amplification. So this mRNA, once it gets inside the cells, it will start re-amplify itself, making more and more copy of the mRNA, and this will result in more protein production if we compare it to the conventional mRNA. Usually we expose our mRNA to different quality controls assay that range from as simple as RNA electrophoresis to chromatography to make sure that we get higher purity integral mRNA. We do also in vitro and in vivo translation to make sure that what is the mRNA that we are producing can be translated to a protein. You skip the slide. So during the COVID-19, all the platform or all the known platform for vaccine development have been utilized, including mRNA. And if we see at the bottom of the table that we have plenty of the mRNA groups that work in development of the mRNA, either in the preclinical trials. We have roughly around 21 mRNA, and we have now almost 6 different mRNA in the clinical trials. So for our work, we use the previous platform using the international sequence. However, we went back and analyzed all the sequence of the coronavirus, SARS-2 coronavirus from Saudi Arabia, because we want to base our vaccine in the Saudi sequence and neighboring countries. So there is around 558 sequence published from Saudi Arabia. We take all these sequence and analyze them, and we didn't see any difference between them and the most frequent sequence of circulating virus worldwide. So the first sequence is 1383, which is represent around 61 worldwide, and this sequence represents around 75% of the circulating viruses in Saudi Arabia. The other sequence is sequence 399, which is represent 16% worldwide and represented around 26% in Saudi Arabia. So we take this sequence and based our vaccine on them. So because we are using two platforms, either the conventional mRNA platform or self-replicating mRNA platform, we start with the sequence 1383 and either use the full spike protein or the RBD domain of the spike protein, and that generates for us 4 different candidates. And since the spike protein between these two circulating viruses are similar, so for sequence 399, we didn't use the RBD domain since it is similar to the sequence 1383. So we just use the whole sequence of the spike protein from this sequence, either in the self-replicating enzymes or in the full spike protein enzymes.
You have 2 minutes.
Sure. So this is the overall workflow. So we have a continuous our effort to check the sequenced virus from Saudi Arabia, if there is any mutation, that we can include this sequence in our platform and then go to the antigen design. And similar to what I said previously, we do the cloning, amplifications, and then the in vitro translation of our mRNA. Now we are in the process for making in vitro mRNA, and we analyze it using either electrophoresis or different quality control assays of this purified mRNA, and hopefully, inshallah, within a couple months, we start doing the animal studies. Okay, just the last slide. Okay. Yeah, with that, I would like to thank. Slide before.
Yes.
Okay. Okay. With that, I would like to thank my group at King Saud University, and I want to thank our collaborator at KMARC, Dr. Majid Al-Gharibi and Dr. Michelle, Dr. Lilian, for their support during this work. Special thanks to our sponsor of this project, KMARC, and other sponsor for other project and our international collaborator at Baylor College of Medicine. Thank you very much.
Thank you very much, Dr. Mishal, and thank you very much all the speakers. Now we will have around 15 minutes for discussion. So I would like to start this session, the Q&A, with the first question, and I would like to address it to all. What is the percentage, the possibility to develop second infection after 70 days recovery after the first dose? If anyone can elaborate on this.
Yeah, I'll take that. Actually, there is no percentage of the reinfection. We do know it exists, but I think it's still rare. So after 70 days, there have been reports that reinfection can happen after 3 months, 4 months. So I think it happens, but it's still rare. And I think also the antibody can, if it does not protect, it can make the second infection less severe.
Okay.
Yeah, if I may add to that, yes. I think it's a rare event. I think only a few cases being reported so far, and majority of people actually develop antibodies, and they sustain the antibody levels in the serum, and most importantly, they maintain memory cells that could be recalled with any reinfection. I don't believe any of the cases have been looked at very carefully with the reinfection, if they have actually developed antibodies at the first place. If most of them were asymptomatic, and I think, as we saw in the presentation by Abdullah and maybe others also already published, asymptomatic and mild infection usually tend to have, or maybe asymptomatic infections that they show no symptoms. Maybe they don't even develop antibodies. So I think that's an important point we have to consider when we thinking reinfection. Memory is good, is the defender of our immune system and our bodies. So I think we will be hopefully very well protected with the vaccine that is coming soon, inshallah.
Great. In light of this question, I have another question from the audience, and this is related to the age. Probably Dr. Naif might answer this question. Is there a differences in the response with regard to the, or correlate with the age? Talking about the three main groups, the children, the young people, the normal generation, and the elderly people. Do you expect to see any response, immune response, with regards to the correlate with the age?
Well, I think that's why it's important to evaluate vaccine in elder people, or at least I wouldn't say elder, but more than 65 years old, because you would expect that the immune response is exhausted by this age, and you may not induce strong antibody. However, we've seen data yesterday from some of the vaccine development that they're seeing a good immune response above 65 years old.
What about children? The small, young age group?
I don't think vaccine people evaluated less than 18 years old. I'm not aware of people start, but that's why we don't know yet the answer.
Any prediction, any expectations?
Well,
that you might
I think the infection seems to induce response in children, so it is expected to see vaccine good response in children, but it has to be done, I mean, in terms of vaccines.
And the other thing, like, related to the first question, like when we say reinfection, we know SARS-2 is a lower respiratory infection as opposed to the common cold seasonal human coronavirus, which is an upper respiratory infection. So for the seasonal coronaviruses, we get them every year, more or less, which indicates the immune response generated by this year infection would wane over a year. So we know now that the immune response drop after 3 months, start declining after 3 months. But maybe related to Anwar's point where we say we're going to be protected, I think yes, but we're talking about now about here. Any vaccine that could give you a good immune response for 1 year, that would be able to break the chain and change the transmission, the epidemiology of the COVID-19.
So do you think, Dr. Naif, that any type of vaccines that was mentioned by Dr. Mashal in that NICE table would require a booster dose after like 1 year?
Yes. Well, I think so because of one reason. We now see vaccines being developed as prime and boost. So if you look at Moderna, they're giving 2 doses. If you look at Oxford now, they're thinking 2 doses. So people are already thinking of 2 doses vaccine. For sure, you would need more boost over the years. But in my personal opinion, I think the vaccine is needed during the pandemic, not to eliminate the disease completely, but to break the chain of transmission, to reduce, to put it under control. And over the year, you could vaccinate further people as you go along. Because it's not going to be a miracle answer. You give the vaccine to the whole world and that's it. You wipe out the virus. It just like you contribute to more controlling the virus.
Probably the next question is Dr. Mashal. By the way, Dr. Mashal, if you can stop sharing the screen so we can see everybody's here. The question to you, Dr. Mashal, is about the expected side effect, and we have seen some serious side effects happen with some of the vaccines in phase 3 clinical trial. So, what do you expect, or what is your prediction of a severe side effect that we might expect from different type of vaccines, starting with the mRNA vaccine?
Yeah. So regarding the safety profile, most of the clinical studies show us that most of the vaccine now there is some cases, but we cannot really link it directly to the vaccines. However, I think during this pandemic, since there is a fast development and there is a speed up in the clinical trial, we might didn't find some side effect appear now, but it will appear in the long term. One of the possibility, because most of the newly developed or used platform include vector-based vaccines, DNA-based vaccines, or RNA-based vaccines. We don't have any example of this platform in the clinic. So that really, let's say, delay their development during the last couple of years. But I think this pandemic facilitate the further development of this platform. But to be honest, we don't know about the safety for a long term about these vaccines. in my opinion, maybe mRNA, since they are more transient character and they are not stable for a long time inside the cells, maybe we will not absorb. So, and that can also happen with other blood forms. But let's say until now we didn't see something serious about the side effect or the safety. But we know that even after the vaccine launching, we'll have the phase 4, which is surveillance for any side effects. So if we can see anything after launching, and that possibly could happen with these kind of the new platforms, they can be stopped or withdrawn from the market.
Absolutely. Most of the vaccines in phase 3 now, they are testing the vaccine on thousands of people. Sure. It's not like hundreds. So hopefully we will see more reports if there's any concern about these vaccines. With moving to the not the vaccine, but a question, in fact, that comes in everybody's mind, which is a couple of countries, they reported a reinfection. Can anyone elaborate on this? And would that be a concern for the vaccine? If there is immune response developed after the first infection, then why people get another infection maybe after a few months?
I might start, I think, and maybe Naif or Abdullah can take over. But I think the brilliant presentation by Bali, Plunder, and Wissel showed how people could respond differently to everything. Vaccine, we're talking today about precision, personalized medicine. And I think it's important to understand that with regards to everything. I think Bali has been working on this for a long time, and we were seeing it today. In reality, this is the first pandemic or new virus that we observe at this scale in the current population of the world. Maybe 1 or 2 persons not today experienced 1918 pandemic and this pandemic. So we're gonna see everything. We're gonna see the reality of every single the new infection of the new virus emergence and outcomes. Again, as we mentioned and Abdullah showed before, not everybody is responding. And we don't know if these people actually responded well at the first place, or they were just mechanically infected. They were just positive because of RNA. I haven't seen any severe case so far that is, I don't know, maybe someone saw something similar. Severe case that got reinfected. Most of the cases, I think, they were mild or asymptomatic and they got reinfected. And maybe Naif or Abdullah.
Yes, I think it's a complicated topic here, like, because we're talking about reinfection, and if we rule out all these false positive and false negative testing and all that, and now we know like reinfection is happening. Yes, it's very rare, but it is happening. And I think multiple points here. The first point is the immune response will wane over time, and we know to start with the first infection, if it's severe, you get higher antibody. And that's what Abdullah showed today and other people internationally showed. The severe the case, the more antibody you get. So if you had the first infection is not severe, then it's less likely that, if the first infection is severe, it's less likely to get another infection. If it's mild or asymptomatic, you're more likely to get another infection because you did not induce antibody strong enough. And that antibody, even if you induced a little bit of antibody levels, these would wane over time. And again, for the vaccine, I don't think it's a counter issue for vaccine development. Yes, we know it's going to happen, the reinfection is going to happen, but that's why we need the vaccine to contribute to more antibody being, like more people being seropositive, basically. And I can hire an example from our camel work and other people. Bali yesterday also talked about this, that the memory is what it counts. Sometimes you need to induce some memory. So next infection is important to count to be more protective. He showed yesterday that we might also see some memory innate immune response. And in the camel study, we saw people come out getting reinfection again and again, but when you vaccinate them, then you raise the level of antibody to a very high level that protect reinfection. Thank you.
It makes sense. Since we have you, Dr. Naif, can you? I got a question from the chat. Are you using any adjuvant in the ChAdOx vaccine?
Well, we don't use, usually, a rule of thumb, you don't use adjuvant for viral vectors because viral vector by themselves are having adjuvant capability.
Okay. Well, more questions, in fact, and concern about the when do we expect to have the vaccines ready and available for use? I know there is a competition, but after maybe the last serious reports, I think some of the companies slow down and maybe have more robust, I guess, monitoring system for the adverse events. So, what is your expectations for the first vaccine to be available?
If you allow me to start. So regarding this, I think now because we have different platform and different vaccine, the phase 3 clinical trial, which is very good. That means that at least we will have a couple of them launched in the market. The thing is, I am mainly concerned about the manufacturing and when we'll get the vaccine themselves. Like I know that in different countries they already have vaccine from the company that working for them. So I will say like early next year, maybe we'll have a couple of them, at least 1 or 2 vaccine in the market. But these vaccine also, there is a question about their efficacy and are they the best vaccines? In my opinion, and I was discussing with different people, with Dr. Peter Hotes, that usually the best vaccine will come later.
All right. Do you expect efficacy of a vaccine for a person who got infected? And do you think there will be a difference in the immune response or the immunity itself for somebody who got infected and get the vaccine? Is it going to boost their immunity, or is it going to be, I mean, the same, or what's your opinion on that?
I think is a question general or Dr.
Oh, I think general, yeah, because
I think the best guy to answer maybe Abdullah, because he's worked on this enhanced antibody disease. But usually the concern is if you vaccinate and then you get exposed to the natural infection, not the other way around. Because if you get if you get exposed to the virus naturally, you induce antibody response and immunity to all the protein and antigens. Then the vaccine will just act as a booster to a single antigen, which is the spike in most cases. But I will leave it to Abdullah.
Actually, I did not catch the question, but I think it's regarding the immunopathology of vaccines, right? So for SARS-CoV-2, I think the answer is we don't know. We haven't vaccinated people in large scale and then exposed them to the infection, so we don't know. And that's the same thing with the SARS-CoV-1, with MERS. We don't know if vaccinated humans can also be exposed to the natural infection, can have immunopathology reaction. But based on some experience in other respiratory viruses that really rare, and most of the vaccine now test this early on during their preclinical or clinical trials. So I don't think it's an issue, but we don't know actually the right answer.
Well, I think let's hope we get enough vaccines for people who are not infected at all.
High risk, not infected people.
And then we'll think about that.
Yeah, sure.
I think to add something on the vaccine platform that is going to be used. So certain vaccines elicit humoral antibody responses only, while others elicit both humoral and cellular, like the live attenuated vaccine. So it also depends on the vaccine that you're going to take. So some of those vaccines will mimic natural, since you're giving them the whole virus, while others you don't expect high levels of immune responses boosted. So it's also comparable not only or out, but also the platform technology that is used for testing.
Sure. Well, with that, I would like to thank you all for your outstanding, great presentations and your contribution. Thank you very much, and hope to see you next conference. Thank you all.
Thank you very much.
Thank you very much. Thank you.
Thank you.
Bye-bye.
We now begin at one time and with one goal. A journey on a bullet train in which we race against time towards developing a vaccine to save life and humanity. This journey includes 32 experts and 10 scientific sessions, in which we discuss 6 topics revolving around developing a successful vaccine against the COVID-19 virus. From Riyadh, Kingdom of Saudi Arabia, we welcome experts to whom the world looks with hope, and major companies that will lead the way in distributing vaccines around the world. From Health Affairs of Ministry of National Guard, King Abdullah International Medical Research Center, KMARC, King Saud bin Abdulaziz University for Health Sciences, and the Saudi Secretariat of the Group of 20, G20. We say to everyone, let us proceed with one ambition and with one goal towards a meaningful life. And let us work together to protect the health of our fellow human beings and our future generations.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound, where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافت قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound, where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
مملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound. Where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافت قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
From Riyadh, Kingdom of Saudi Arabia, I would like to welcome you all and thank you for attending this session. This session will be about accessibility to COVID-19 vaccine. Today, I have three distinguished speakers who are going to enrich this session and elaborate more on its title. Our first speaker is Mr. Richard Wilder. Mr. Wilder is the General Counsel and Director of Business Development at the Coalition of Epidemic Preparedness Innovations, CEPI. There, he and his team are responsible for drafting and negotiating legal instruments necessary to progress CEPI mission and support its administrative requirements. Mr. Wilder spent years as a partner in a global law firm where he specialized in international trade law and practiced in the field of global public health. Please join me in welcoming Mr. Wilder.
Thank you very, very much, Dr. Ahmedjad. It's a great pleasure for me to be here today, to have an opportunity to participate in this seminar on COVID-19 vaccines, global challenges and prospects. Today we're going to be talking about accessibility to the vaccine, and I'm going to focus in particular on CEPI, my organization, that is the Coalition for Epidemic Preparedness Innovations, and the COVAX initiative to advance vaccine accessibility. The overview of the presentation is quite simple. I'm going to give an introduction to the ACT Accelerator, and I'll explain what that is in just a moment, and to COVAX itself, and an introduction to CEPI, its history, and our work on COVAX, and then focus on status and operational details that relate to the core issue here of access to the vaccines once they are developed. First of all, starting with the overarching organization, is the Access to COVID-19 Tools Accelerator, or the ACT Accelerator. This was conceived just during the span of this year with the outbreak of COVID-19, or SARS-CoV-2, the virus that actually causes COVID-19. We recognized early on that the only way for the work to succeed in terms of developing the necessary tools to deal with the crisis, to deal with the pandemic, is to join together. So as you can see, there's a very large number of organizations that have come together to work on several pillars or several areas of focus that are necessary to address this pandemic that arose in the early part of this year, late last year, and unfortunately is once again getting worse. So there's pillars related to access and allocation, how one goes about deciding once vaccines or drugs or diagnostics are developed, how it is that they're going to be made accessible on a global basis. The development of vaccines and their access, and that's the area that we have specific responsibility for as CEPI. Therapeutics or drugs, and the Wellcome Trust and Unitaid are involved in that effort. The World Bank funding the improvement of health systems, and the Global Fund and Find, the organization that specifically deals with diagnostics, have a focus on diagnostics. So I wanted to give you that overall picture of the ACT Accelerator before we focus specifically on vaccines and COVAX. So the COVAX vaccine pillar of the ACT Accelerator includes three principal actors, CEPI, of course, Gavi, the Vaccine Alliance, and the World Health Organization. And just as was the case with the ACT Accelerator, it's not possible to undertake to do the broad work that's required without working in close cooperation with other organizations that have specific and important areas of responsibility. So from CEPI's perspective, our focus is on supporting vaccine research and development, taking vaccines from an early stage idea in the lab all the way through clinical trials and into production. Gavi has been an organization in existence for a number of years and has a responsibility for obtaining or attracting the funding necessary to fund the procurement of vaccines and to ensure that there's a broad supply of safe and effective vaccines available, and then to fund their allocation. The World Health Organization, in the context we're talking about today, they're providing guidance to their member states on vaccine policy, safety, and regulation. And importantly, they have a role in setting the algorithm or the mechanism whereby the vaccines, once developed, are going to be made accessible. Turning to CEPI specifically, is that our vision is a world where epidemics are no longer a threat to humanity, and we accelerate the development of vaccines against emerging infectious diseases. We're actually a very young organization. We were launched at the World Economic Forum in 2017, so we only have an existence of a little over 3 years, actually, coming on 4. And since that time, we've undertaken to do work in a number of areas where there's potential epidemic threats. We're obviously focused on a pandemic now with COVID-19, but at the beginning, our focus was on epidemics, recognizing that epidemics affect us all. And there's a number of different components to responding to epidemics that I think are important. One is the paradox of progress, in the sense that we now have much greater global trade, we have dead cities, we have easy travel. Ecological change means that infectious disease have easier means to travel around the world rapidly, as we've seen with this example of COVID-19. The harm is immediate and long-lasting. It can disrupt travel, it can disrupt business. And again, I don't have to explain that to you. We are seeing it play out before our eyes with COVID-19. and the costs are high in terms of the human costs of people falling ill and dying, and economic costs both at the personal level to individuals and their loss of income, to the local levels with businesses finding a difficult time to thrive and survive, and through to economies around the world. So initially, our focus was on a number of diseases that are on the World Health Organization R&D blueprint list. So these are MERS, Lassa, Nipah, chikungunya, Rift Valley fever. And in connection with those projects, especially with MERS, we had occasion to work with institutions in the Kingdom of Saudi Arabia. And those projects continue. But we also had a disease X, and disease X was the disease that we all knew was going to come, was going to present a problem as an epidemic, a potential pandemic. And of course, we have that now with SARS-CoV-2, the virus that causes COVID-19. So for COVAX, again, it's the element of the ACT accelerator that has responsibility for vaccines, have a number of very ambitious goals. We are in the process of supporting the establishment and the buildup and the maintenance of the largest active portfolio of vaccine candidates globally ever, to deliver 2 billion doses by the end of 2021, and not just deliver them through single channels of distribution to a handful of countries, but on a global basis, and to offer return on investment that's compelling in the sense that, as I mentioned before, with epidemics and with pandemics, the harm that's caused at the local level is significant. And the quicker we can get the vaccines out, the faster we can not only prevent people from falling sick, but also to resuscitate and save the economies around the world. And then guarantee a fair and equitable access to COVID-19 for all, in the sense of no country being left behind. What's happened in the past is vaccines have been developed, and for low and middle income countries, they find themselves getting access to those vaccines 2, 3, 4, 5 years after the wealthier countries in the world get access. And so with COVAX, the goal is to ensure the rollout and accessibility to those vaccines at the same moment around the world, and ultimately to end the acute phase of the pandemic by the end of 2021. So we have a diverse portfolio of candidates across a number of platforms of technology I'll explain in a moment. We have expert industry support across the work of CEPI and across the work of COVAX to be sure that we are getting the best scientific comment and input into our work. We have flexibility to move resources to where they're needed most, and we're continuously assessing our opportunities and actively managing our portfolio. Right now, we are actively developing vaccines in several different areas of technology or platforms. So protein vaccines, nucleic acid, viral vector, and inactivated vaccines. We have now under CEPI, we have 10 vaccines under development and looking at a couple of more. We need 3 to 5 vaccines to actually come through clinical trials and in the market to be able to reach that 2 billion dose goal in 2021. The issue there being, of course, is that the development of vaccines is a scientific undertaking, and there's a fair amount of uncertainty in the processes that one has to go through. So you don't know if a vaccine project, once started, is actually going to go the distance. Will you find a safety signal at some point? Will it show that what you thought might be a highly effective vaccine lacks sufficient efficacy to be used? Or maybe it's a vaccine that requires complicated preparation at the bedside, when the vaccine is administered, that limits its usage in many countries. I'm not going to spend a lot of time on this slide just to lay out the fact that we do have a significant portfolio of vaccines currently under development with companies from around the world and conducting clinical trials around the world. And many of these vaccines are in later stages, phase 3 of clinical trial development, and which means that they're closer to the end, closer to the point where they'll be finally developed and available to the market. We have additional investment priorities because we're constantly revisiting our portfolio, revisiting the individual projects, and looking at additional R&D investments, some of which I've showed here for some vaccines that could be included and brought to market. There's also a so-called wave 2. What I've been discussing is wave 1, so very early candidates, some of which we started on in January of this year, almost immediately after gaining access to the gene sequence for the SARS-CoV-2 virus. We began work to develop vaccines, so we added quickly to that portfolio, and now looking at wave 2 of vaccines that have improved attributes in terms of single dose, perhaps lower cost, ease of administration, a broader range of patients that might be able to have access to them. We're working to basically to develop some additional funding. We on the CEPI side for full development of our portfolio have a funding shortfall of about $0.82 billion, which we're working to shore up in an effort to be sure that we have not only the funds available to fully develop the candidates under development, but also to broaden, the range. So this is, I just have a couple of slides left actually in my presentation. And I wanted to say that I'd like to talk a bit about the back end, if you will, of the COVAX facility. And that is, once you have a vaccine that has been identified for development, it goes through development, clinical trials, and it's shown to be safe and effective. You then, begin the process, or you ramp up production and make it available to the market. What then is done in terms of the allocation of those vaccines on a global basis to address this global pandemic? One thing I should say, before I speak specifically to this slide, is that we are doing things, fundamentally different in the context of ACT Accelerator as well as COVAX. Fundamentally different in a couple senses. The one we talk often about how we're driven by the need to ensure speed, scale, and access. And so we're moving very, very fast with these projects, faster than is normally done, but not fast in a way that compromises safety and efficacy, but fast in the sense that we're doing things in parallel that we would normally do in sequence. So one example is manufacturing, where for our projects, while we are undertaking to do the development work to show the vaccine is safe and effective, we're also beginning the task of building out, scaling up manufacturing capacity, so that the moment that vaccine gets approval, there are doses of vaccine that are ready and available for global distribution, global allocation. And so we're doing that at risk because one would normally wait until the vaccine is developed and bring it into the market. What we're charged to do for COVAX is to accomplish all of what I've described in the span of about a year to 18 months, where the normal cycle for vaccine development is 7 to 10 years. So we're shortening things significantly. Again, not compromising safety, not compromising efficacy, but doing things in parallel and focusing more resources on the tasks to be done. So when the vaccines are available, there are a couple of things that have to be done through the COVAX facility. And the COVAX facility is primarily under the control of Gavi, the Vaccine Alliance. And the things they have to do is to secure the funds that would be necessary to purchase or supplement the purchase price of vaccines for low and middle income countries. So you can see in the middle of this chart, it says AMC donor makes a grant to the Gavi Alliance. The AMC is the advanced market commitment. That's the vehicle, through which low and middle income countries and upper middle income countries can get access to funds to purchase vaccines. We also have countries from high income countries, and they are the self-financing participants into the COVAX facility. So they are providing their own money for the purchase of vaccines. And so what's required is for all of the self-financing participants and the low and middle income countries to make a commitment or make an application for support. So we know the number of countries that are going to be getting vaccines from the COVAX facility. We know the volume of the vaccines that are going to be required, and we can ensure then that the COVAX facility can meet those requirements. And then down below, there's several boxes that talk about a shareholders council, which is made up of the self-financing participants, the advanced market commitment engagement group, that is those that are funded by the COVAX facility, and then a consensus group that takes on specific issues that are looked at by those two groups and comes to a consensus. There's an actual office for the COVAX facility that has the individual people that manage that facility. They will take into consideration the vaccines that are being made available and identified to it through the independent product group and the procurement reference group. And once all of those inputs are in, so we have an identification of all the countries that are participating, both those that are self-funding, those that require funding from Gavi. We have the indication of the readiness of those countries to receive vaccines. We have an identification of the vaccines and the volume that are available. There is then a task force that has the job of the allocation of the vaccines across all of those countries. The goal for allocation is to make sure that all countries, low and middle income countries as well as high income countries, receive vaccines at approximately the same time. It's important, for purposes of equity and safety, that those vaccines are available to all countries, so that you don't have a situation where vaccines, might be available in high-income countries and then some years later to low-income countries. And so we're setting up this facility specifically to ensure that countries across the world will get access to vaccines at the same time, and that the doses that are available are also equitably allocated. The number of doses, there will be doses coming available in early 2021 of some of the vaccines that I've mentioned. They will go through the process of determining safety and efficacy, and they will be available to the market. They will then be available for allocation by the COVAX facility. And the intent of the COVAX facility is to ensure that all countries get vaccines, as early as possible, but they all get a certain number of vaccines that are sufficient to have a public health impact on the COVID outbreak. And so a certain number of a certain percentage of their population that's covered, to address the needs of frontline healthcare workers first, to address those people that are most at risk second, and then to provide for general vaccination of the population at some later date, so that succession of access of volume of vaccines to address specific public health needs. So this is my last slide, and so what I want to do is to go over just a couple of points in conclusion. One is, to make the overarching point that we now have 186 countries around the world that are participating in the COVAX facility. So 92 are the AMC countries. They're receiving support for vaccine procurement from Gavi. There's 94 self-funded economies, those that are paying for the vaccine doses themselves. In order for us to ensure that we have equitable access to vaccines, of course, the first point is that vaccines have to be developed. And so there has been and continues to be need for funding to ensure that we have a very robust platform of vaccines based on a number of different technical platforms that are available for allocation out to the COVAX facility and to the countries. We need necessary funds for the procurement of the Gavi 92 countries. And so that's an ongoing effort as well that Gavi is mentioning, is managing. And then another important point is, and this is something that WHO and Gavi and also UNICEF and others are working on, is to ensure country readiness to receive and use the vaccines, to ensure that there is the necessary facilities to receive and properly store the vaccines, because they do require cold storage for the vaccines, to ensure that the infrastructure exists for the distribution of vaccines out to clinics where they're going to be used, and that the health professionals necessary to administer the vaccines are in place, and the necessary data resources are in place to keep track of the use of the vaccines, to keep track of any instances of adverse events and the like for particular vaccines. So there's a country readiness component to the work of COVAX as well. And then last, once we have the vaccines, the funding is available for the procurement, countries are ready to receive, the vaccines will then be allocated through COVAX to be sure, as I said, no country is left behind, so that we have first doses go out on a global basis, and the volume of allocation is sufficient on a global basis to meet the public health needs as I've described. So thank you very much. I appreciate your attention, and I look forward to any questions that might come. And I think questions are at the end of all the presentations. So thank you very much.
Thank you very much, Mr. Wilder, for this interesting and informative presentation. We'll go for our second speaker, Dr. Marwan Baker. Dr. Baker is the chairman and founder of Bring Hope Humanitarian Foundation. He has been involved in various humanitarian efforts since the late 90s. Dr. Baker is a PhD graduate from Technical University of Denmark, and he has a master's degree in physics and medical physics in cancer radiation therapy. As a personal witness and survivor of the 8-year Iran-Iraq war and the chemical attack on Halabja, the Kuwait War, and the massive Kurdish exodus during the First Gulf War, Dr. Baker sought to always provide assistance to others in need, going to help those struggling, not just the social victims of poverty, but the ill and infirm, and especially vulnerable children and women, people with cancer, or students whose schooling was in jeopardy for the lack of funding. Dr. Baker did all he could to come to the aid of people who were in desperate need in one way or another. Please join me in welcoming Dr. Baker.
Thank you very much, Dr. Faisal. Good morning, good afternoon, good evening, wherever you are in the world. My presentation today will be a little different and general, and it will be some overlap with the former speaker, Mr. Walden, because I didn't see his presentation. And one of the things that I'm going also to take up is personal experiences, because I'm in the humanitarian field, and today my topic will be about challenges in the delivery of the COVID-19 vaccine to people in need. I would like to start with a question to everybody. Imagine that a vaccine with a great efficacy and safe is developed wherever in the world, and I am reaching you and say that, okay, this vaccine is ready. What would you consider to accept a vaccine that will be vaccinating yourself and your family and your kids? I leave this one for you until the end of the presentation. So I hope that you can see my presentation now. I go to the next. So The agenda today is people in need. Who are they? How many of them? Where are they? And I'm also going to talk about in the history of human history, there have been successful and cost-effective global healthcare interventions. What are those? Is vaccine one of them? Also, I'm going to talk about the challenges that we might have or we have with the COVID-19 virus, challenges with the developing COVID-19 vaccine, challenges with the global vaccination to reach each individual on this earth. Why this is matter? Because I'm referring to a quote by Dr. Leong from the Hong Kong University Medical Center. He said that as far as there's a single individual on this earth which has not been vaccinated and cured from infectious COVID-19, wherever you are in the world, you are not entirely safe. And of course, there are also about skepticism and misconception of any vaccine. Previously, we have never experienced something like this. We are so skeptics about that, where this vaccine coming from, and we are making geographically, which is absolutely created confusion about human beings on this earth.
Sorry, Dr. Baker, for interruption. Can you please make your full screen of your presentation, please?
Do you see it now? It's the full one.
Yes, thank you.
Is it okay? Yeah, great. So I would like to just come with the good news, because despite all of these challenges, there are solutions, and that's what I'm going to tell you. So, who are people in need? Some of them are displaced and refugees. Why I put this in the up? There are around 18 million people around the world now who are displaced. And 29 years ago, I was one of them. After Kuwait War, I was displaced, trapped on the mountain between Iran and Iraq. Why I'm taking up the example like this, I will tell you what challenges we have to reach each individual of displaced people. People in need, in conflict zones, war zones, in the remotest areas. There could be some 3 families in some village or 10 families in some other villages. I mean, we have millions of villages across the world which we have difficulty to reach them. Urban slum population, we have approximately 1 billion people living in the slums. It could be in Bangladesh, India, African continent, South America, people in poverty. And they are, of course, overlap. Indigenous people, well, I'm taking up this one because indigenous people are usually not fairly treated, even in the developed countries. I'm based in Sweden, one of the best countries in the world. Still, we have some challenges. Regarding Canada, they are really one of the best countries which are intervening when it's coming to the humanitarian emergency. They are first on the ground, and they are opening the doors for the refugees. But regarding treating their own indigenous people, there are also some challenges there. So all of these leading to that we will get inequality in immunization coverage worldwide, because it's depending on what geography you are based. If you are in the urban and rural areas, the same country, those who are urban, they are usually vaccinated earlier. Socioeconomic status, of course, riches, high educated, usually vaccinated first. What Mr. Walder talking about, that reaching everybody at the same time using the COVAX system is fantastic. Why he's talking about that? Because that has not been implemented before. Usually you come with a vaccine, it takes 2-3 years to get to most of the people in need around the world. And this is leading to one thing. Today, there are millions of children are not vaccinated against common vaccinated preventable diseases. Could be polio, smallpox, measles, and so on. But in human history, we have had a lot of successful and cost-effective global healthcare interventions also. Vaccine has been one of them against infectious diseases. Actually, sanitation had been as effective or even more sometimes with the clean water and sewage disposal, but still there are millions of people who are lacking of clean water and sewage disposal. General hygiene, very important. At Bring Hope Humanitarian Foundation, we have been able to mobilize millions of hygiene items around the world, in Middle East and Africa especially. Why this is important? Because even if it's coming to the infectious diseases and contagious viruses like COVID-19, what we do? Sanitizer, washing your hands, and clean everywhere, surfaces, and so on. So hygiene has been really, really important to prevent spreading of diseases or stopping it. Antibiotic and antiseptics have been great things having happened in the human history. Other treatments. Imagine that for other treatments, we are spending trillions of dollars. It could be medical equipment and medical drugs. But if only a portion of that one have been used for developing vaccine, the last 2-3 decades, or focusing on the sanitation for the people in need in low-income countries, or awareness about how important is the hygiene, actually we could stop some diseases to spread. So probably this is a wake-up call that nobody in the world has been taking care of hygiene as we are doing it right now. But not everything is black. The last 4 decades in the world, there have been extensive global health improvements, especially in the low-income countries. And therefore, this has led to longer life expectancy worldwide, but still many countries suffer. So what challenge we have with this virus? It's actually the worst pandemic, or one of the worst pandemic in modern human history, extremely contagious. We have observed mutations, and this is actually could be a big problem because a developed vaccine could be modified depending on mutation. Chronic disabilities in different organs have been observed. It could be kidneys, liver, lungs, or muscle tissues. Unfortunately, also brain, but this is too early to conclude anything. Also, we have seen reinfections, not many of them, but sadly this has been also observed, but it's too early to conclude anything. Age-related risk categories, we know that elderly are more susceptible to this virus. Challenges with the vaccine. Why do we need vaccine? Vaccine is needed to get a population immunity, or it's called for herd immunity, but this vaccine should have some efficacy. Unfortunately, to get 100% efficacy is not possible. What is being discussed and shown that 50% and over is needed to get population immunity. In Sweden, when I'm based at little Sweden with 10 million people, we have been focusing, it has never been locked down here during the period. We thought that we will get a population immunity just by allowing it to go around. But of course, social distancing, we are following this great nation awareness of the disease, and they are taking care of themselves. But it's very difficult to get herd immunity if you are not having a vaccine. So let's hope that we get a vaccine that hopefully will be up to 60% efficacy. Under 50%, it will be a little difficult and takes time. Safety, adverse side effects, it's too early to know about the vaccine. Age-related effectiveness, we don't know. Cost, yesterday we heard that it can cost one dose $10 to $20. It depends on the scale that it can go down if it's in the bigger scale. Booster doses is really difficult because if you have difficulty to reach people in need with the first dose, then you have risk to miss millions of them for the second. Cold chain, this is how it is also difficult, and the challenge, big challenge, because how can you keep this vaccine, under control in the freezer and just get into all the remotest areas everywhere in the world? Immunity duration, we don't know. unsafe injections using syringe or needle. It's actually have been shown earlier also that disease can be spread by using syringe. But let me tell you also one more thing about syringe and needles for the people in need in local countries. Let's go back to when I was displaced, trapped on the mountain between Iran and Iraq. The sky was our roof, no food, people dying around me, and the group from WHO reaching us with a needle and vaccinating me and my kids against any disease. People say that the risk is that you are refusing. We don't have shelter. There's no food. And there's people who are dying because of hunger or because of anything else. And you are coming with a syringe and injecting, and there are people who will be actually refusing that. And while I'm talking to you about that, we have millions of displaced people in such a situation. How can we convince them to accept any vaccine? Challenges to deliver vaccine to the people in need. People in need usually are in those countries which have suffering from poor healthcare system. They cannot afford for it. That's why it's really great with the COVAX that are built cover vaccine. Limited human resource capacity. I mean, this is to reach all the small villages everywhere in the world. You really need it, and they are lacking of that. Political and social instabilities. Previously, the healthcare workers who have been vaccinating people, for instance, in Afghanistan, in Yemen, in Syria, in Iraq, they are facing difficulties because of political and social instabilities. Customs clearance. Bring Hope Humanitarian Foundation have been mobilizing hundreds of containers around the world, mostly donations that we have received from corporations. And it have been generous people and corporations who have helped with the logistics and covering the transportation also. Finding reputable partners on the ground who have infrastructure to distribute goods, especially hygiene items. All of these are challenges and you do it, but the most biggest challenge is when the container arrives into the country, the customs clearance which are taking a long time, or it's a headache. How can we be sure that if we are sending 300 million doses to Nigeria, that things go smoothly? And this is one of the challenges that should be taken into consideration. And then we have other challenges also regarding vaccine delivery to the people in need. Because those countries, they don't have sufficient supply chain, logistic infrastructure, bad road, running water, electricity, telephone coverage, and so on, warehousing, inadequate cold chain equipment, and then also data monitoring. Because if it's the 2 doses, we have to record and keep track on every vaccination for every individual. But luckily, the IT world has been developed so much the last 2 decades. I think that we can manage that better now. Population growth. I am 47 years old. 47 years ago, we were half of the population as we are right now, almost. It means that any vaccination, global vaccination 47 years ago, compared to now, we have double number of the people to take care of, especially if it's some similar things like COVID-19, that everybody should be vaccinated. Do you remember I asked you a question if I'm coming with the vaccine? Would you accept it or not? Why? Because it's a question of the credibility. It's a question of the skepticism and hesitancy, especially this COVID-19, what we have experienced the last 10 months. It's just news coming all over the world. And should I believe in that one or that one? And we have become so confused that we are just concluding things based on where the vaccines and what country coming from. And this is really not good. But before that, there were other challenges in the developed, less developed countries or developing countries. The cultural perceptions for vaccine is really hard because warlords and landlords can stop just that we are not going to allow you to get into the population and vaccinate. Tribal and religious leaders playing a major role in many places, areas in the world. If you are going to Afghanistan, some province, and the tribal leaders say that no, then it's no, it's not possible to get it. And this problem existing everywhere. Look at the Nigeria, how many tribes, how many languages exist. But these are all difficulties that we are facing. But the question is that, is there any solution for that? The answer is yes. The key solution that Mr. Walder has been already discussing and told you is about collaborations. And it's also not only about that, it's about kindness and generosity. Collaboration from the vaccine developers to the manufacturers, governments, financiers who are financing these vaccines, public and private deliverers, and intergovernmental like WHO, UNICEF, Gavi, and so on. Because I've read some articles some years ago, before COVID-19, 2 years ago, WHO, UNICEF, and Gavi were complaining that they, of 30 billion, they have only $20 billion because they couldn't get the proper vaccination of polio somewhere worldwide. $10 billion was huge money to get it from the government. But imagine the last 10, 11 months, more than $10 trillion has been used for controlling this COVID-19. So money existing. The question is that they prioritize. And also, you have to collaborate with the local leaders, local logistic partners, local government and ministers of health. You have to involve them. When you are involving them, it's easier. They can deliver. The trust will be there. Generosity is the key. As I said that we have already mobilized millions of hygiene items and continue right now where I'm talking to you. We have 4 or 5 million hygiene items from Dubai to send it to Africa and Middle East. But we are not paying for it. Says the corporation can donate these vaccines or hygiene items. Manufacturers can also. Logistic companies. For instance, we are talking about a cold chain. Okay, let's say that FedEx say that the entire African continent last coming 2 years, we are going to help with the vaccine delivery. And it is possible. But the question is that if they are ready to do that, and it is, if it's generosity there, believe me that we are going to save a lot of lives and we are saving more monies. Governmental organizations, absolutely, and financial support, because we need people also to support with exactly what Mr. Wallace said, that regarding Gavi is that they are depending on the donations. If it's from government, if it's from corporations or financiers, to helping the people in need. Before stopping my presentation, I would like to give a personal story. Last week, around the dinner table, one of my 2 kids asked me a question. It was nice. I said, Papa, which means dad in Swedish. I said, yeah. Were Adam and Eve Swedish? Adam and Eve Swedish? While they were eagerly waiting for a response from me, I thought about giving a wise answer. I said, no, Adam and Eve, they were not Swedish. They were human beings. And they say that, were they from Sweden then? I said, no, Sweden didn't exist that time. Were they Kurdish from Kurdistan? I said that, no, Kurdistan didn't exist that time. By the way, Kurdistan doesn't exist on the map still. I said that they were human beings. And I want to tell everybody, before any nation or any country, we are human beings. Before any groups and any religions, we are human beings. And if you want to combat global crisis and such pandemic, we have to take humanity as a base for action. Thanks for your attention.
Thank you very much, Dr. Baker. Our third speaker is Dr. Holger Kandler. Kanzler is the Senior Program Officer at Bill and Melinda Gates Foundation. He's an accomplished immunology research scientist and program leader with 15 years of industry experience in drug development, with focus on immune modulation and vaccines. He is a drug discovery expert in innate immunity, vaccines, immune oncology, and viral immunology. Please join me in welcoming Dr. Kanzler.
Good morning, good afternoon, everyone. I hope you can hear me okay. I'm delighted to be invited to this seminar, and I really appreciate the thoughtful and in-depth presentation by the previous presenters, and thank you for the moderator for inviting me. So I'm working at the Bill and Melinda Gates Foundation. I hope you can see my slides in full screen mode. If not, let me know. I'm actually in the what we have built, the COVID vaccine discovery team. And I'm actually presenting here a number of thoughts for the overall team. What I will try to do today is give a bit of a perspective of the early elements in thinking through a vaccine pipeline that would be amenable for use in particular low and middle income countries. So I'm going to take a slightly different perspective in the beginning, but then hopefully at the end of the presentation, I will also arrive at the global distribution challenge that we all want to discuss further today. First of all,
for interruption, can you please full screen your presentation, please?
Let me see how I can do this. Is this better for you?
No, it's still not full screen. Yeah.
But do you see the slides progressing?
Yes, that's okay. Better. Thank you.
I apologize. I don't know. So I will decrease the size here. So it's important that you see the slides progressing. If you don't, then let me know. So the Bill and Melinda Gates Foundation in January actually has decided to tackle COVID-19 with 4 goals in mind. So they're illustrated in the slide here. The first one is really to protect the most vulnerable in that regard. And that really is around the strength and the capability to actually coordinate and detect and respond to COVID-19 in key countries. That's one part of the strategy. The other one is obviously acceleration of detection and containment of the virus globally. The third one that will be of greater relevance today is actually the development of treatment of vaccines. And what we will discuss today is really leveraging the capacity to coordinate and fill certain gaps in the COVID-19 product development, and how the foundation has been trying to contribute to that while securing actually the equitable global access. The other one is obviously minimizing the social and economic impact that this pandemic is posing to all of us. The approach usually has 3 prongs. This is not much different going after other global health challenges in COVID-19. So there's obviously the ability to test, i.e. to identify and develop diagnostics that help us to detect cases of COVID-19 and understand the epi of this new challenge. The next column then is obviously treatment, and what we're focusing on is the development of treatments of mild to moderate disease by small drugs and antibody approaches. And then obviously the thought of preventing further disease by really developing a number of vaccine candidates and make them available for global use. This is obviously a continuum, so as soon as we're done with a safe and effective vaccine, the circle starts again, because development of more efficient testing and treatment will be essential to really get a grip on this rather scary pandemic. One starting point for actually switching over through how to make a vaccine is actually to sit down and develop an intervention target product profile for a COVID-19 vaccine. So obviously this table was not present on the 1st of January. We actually thought this through, and we're sharing this with our grantees and collaborators to illustrate what we think are the key parameters of prophylactic vaccines that would be for general use. So obviously this is divided up in minimum and optimistic product characteristics, and then a number of annotations. And from this document, I took a few key elements that would be important to remember. So we're working on active immunization, and the target population in minimum is really adults, including elderly. However, what we need in the end for broader and quicker global health impact is really vaccines that work across all ages, that work for pregnant women. So these are populations that often in the first wave of vaccines are actually not optimally addressed. In terms of efficacy, a 70% efficacious vaccine would be highly desirable. The higher we can get with the efficacy, the more promising the vaccine overall will be. In terms of the immunogenicity, durable neutralizing antibody responses really seem to be absolutely required. We are certainly looking for additional biomarkers and in particular correlates of protection that help us build these vaccines more effectively. Duration of protection, I think starting with a minimum of 1 year is reasonable. However, the durability of the vaccines and the necessity to really approach this with prime boost regimen needs to be investigated, because in the end we want to have long-term protection. The presentation could be multi-vial in the beginning or then single vial for later application. An important element to really consider is stability of these vaccines, because vaccine technologies actually differ in their requirement in terms of thermostability. We realized from the beginning that the first wave of vaccines actually will have vaccine candidates that require short-term and long-term storage that is actually ultra cold chain. So it is difficult to implement, but it's not impossible. So we actually generated some flexibility in terms of the thermostability requirement, in particular for the first wave of vaccines in this target TPP. Now I'll switch a little bit to the discovery group itself, which is led by Linda Stewart. I'm representing this group today. I'll let you know who actually works on this. And as a background here, the foundation is really heavily investing in the vaccine, is continuing to do so as a public health tool with the greatest impact. Billions have been spent in the last 20 years. And every year, the foundation spends around 50 million really on the discovery of novel vaccines and also monoclonal antibodies across global health indications. When we make these investments, we really often do them in a disease agnostic way. We're very interested in building framework that actually can support the research and development for a number of foundation focus needs and priorities. We are actually very much in favor of prioritizing platform approaches that could be, again, applied across the portfolio rather than disease-specific solutions. And we're very comfortable with working with programs that are long timelines, and we also like to really bet on novel technologies and innovative solutions. For example, we have been active in the field of mRNA vaccines for a long time. The initiatives are broken up in distinct elements. So there are activities that are around subunits plus adjuvant vaccines. There are activities around nucleic acid vaccines, and then also viral vector vaccines. We have a distinct program that is biologics, which is really monoclonal antibody for disease prevention and treatment. We are also investing in correlates, and we have also links over to livestock vaccines. All of this is actually also done in the presence of what we call the Global Health Vaccine Accelerator Platform, which supplies a number of cross-platform technologies that helps to evaluate vaccine candidates and push them forward. For COVID-19 specifically, within the first 30 days of SARS-CoV-2 being identified as the causative agent, we really launched a full vaccine discovery program in that regard. And our complement was really to look at the deficiencies of the vaccine development from a global health perspective, realizing that the initial wave was really focused on providing vaccines at speed and not necessarily at scale. So we really set out to develop a portfolio of wave 2 vaccines that are focused on affordable and scalable solutions that are actually suitable for implementation in low and middle income countries. Obviously, we're not doing this alone. Nothing can be done alone here. Everything is done in collaboration, and we greatly enjoy going in this field with partners, and CEPI is one of the very important ones here. Again, we're mobilizing our existing partners, and we're also really mobilizing manufacturing facilities that are in developing countries, in particular using existing manufacturing capabilities to bring vaccines forward quickly. So how does the foundation do that? And what I will try today is to give you examples of each of these, if you will, points of vaccine discovery, which really goes from understanding of the basis of the virus over to plans to distribute the vaccines using existing networks. Now, stepping back a moment here, if you look at the way that vaccine development has classically been done, and realizing that we're trying to do things quite differently for COVID-19, the vaccine discovery takes time. So the time frame for beginning a vaccine discovery effort in 2020, I think, post-license would be a 2030, 2035 timeframe. Obviously, we cannot afford that. So we challenge ourselves, how can we actually condense that particular timeline to get to vaccines faster? So unfortunately, the slide build doesn't work, so I will improvise here. So what we're trying to get at is basically the timeline down here, where in 12 to 18 months, there's actually a target for vaccine delivery 12 to 18 months after program start. So this is an ambitious goal, but I think the whole world is facing this, and it can be implemented. It really depends on the vaccine platforms. But the challenges that we're facing is that vaccine development is actually quite hard. The probability of success at the different stages of vaccine development is rather low, and we really have to address that from the perspective of global partners, and really realizing that for this particular challenge, novel platforms should be considered, and that we actually need to develop rapidly and simultaneously a portfolio of vaccine platforms, because we will have vaccine winners, but we will also have vaccines that are simply not going to get us to the goal. What I will do in the following few slides is really focus on the early part of this overall vaccine discovery and development process, which is discovery and preclinical and some of the process development. And then in the end, I'd like to end up with some thoughts on how to really get to the billion dose scale that we all will require. So very early, and the following slides are illustrating some of the investments that we made, some of the partners that we're working with. And I should say that we are the funders, we're contributing, but I cannot emphasize enough how much we appreciate the work of the grantees and the collaborators. But in terms of understanding the basics of the vaccine, so how would you build the vaccine? Structural biology of SARS-CoV-2 has been very important. So the coronavirus sequences have become available early in January, and then it's also very clear that the most likely protein to go after is really the spike protein of SARS-CoV-2, which has been resolved with high resolution based on structure-based analysis. And many vaccine candidates, based on those knowledge, are based on a certain stabilized spike protein. So that is what went into the first wave of vaccines and illustrated in the publications over here. However, we're actively interested in a second generation of immunogens. The reason for that is that we're hoping to increase the immunogenicity of the vaccines by this way. That's one of the reasons, which could lead to an eventual dose sparing of the immunogen. Again, that plays into scale and then eventually distribution in larger scale. The other element that was really important in understanding the basics is the identification of antibodies that are neutralizing SARS-CoV-2. A very active program, absolutely impressive how quickly the world actually has generated such antibodies, neutralizing antibodies, and they're really important for two reasons. First of all, they're important to really design structure-based vaccine immunogen candidates. Again, improvement of potency is quite important there. But then also from a foundation perspective and many others, the application of those anti-SARS-CoV-2 brought to neutralizing antibodies as actually therapies is something that very rapidly has been translating into the clinic. And as you know from press releases by companies involved in this, there are now a number of monoclonal antibodies as therapies in clinical development. In terms of leveraging platform, I think one thing that we can mention is that the foundation has been investing in nucleic acid-based vaccine for a long time, a very compelling technology. The pros of this technology is really the ability to mount a very rapid response, versatility in terms of the immunogen design and implication of different immunogens, and also just the potential of being a disruptive technology in terms of small footprint required. A number of challenges. There is not yet a licensed product for an mRNA vaccine, and while it has been emerging drastically in the last half year, I think it's still fair to say that there are slight unknowns about the dose and scalability of these approaches. Nevertheless, mRNA vaccines for COVID-19 are in phase 3 trials, and I'm pretty sure that other mRNA vaccines will follow in that regard. Another platform that we're really considering is the antigen display. So if you are able to present your immunogen against which you want to induce a response in the multivariate display by a nanoparticle, you are actually in a position where you can generate a higher immunogenic product. Again, this has then to play with dose sparing and maybe with a reduction of the use of adjuvants that you need for subunit vaccines. So we have been actively investing in those, and actually recombinant protein vaccines plus adjuvants are really constituting the majority of the wave 2 pipeline that we're building together with others. What's also important when one goes to those particle space is the question of adjuvant access. So we are very interested in ensuring the access to commercial adjuvants for COVID-19 vaccines. A few are listed here, but we also have an active program discovering new adjuvants to increase the number of doses and the accessibility for adjuvant vaccines globally. What you also need to do, you need to build a structure to really understand the immunogenicity of these vaccines, and again, in collaboration. Mice are often used for immunogenicity, but what's also important is that we have made a number of investments really trying to help build models that help us understand the early efficacy of vaccines in rodents and non-human primates, and also models to really understand the potential safety risk that might potentially be posed for some of these vaccines while or after they have been going to the clinic. So all of this is actually supported by the Global Health Vaccine Accelerator platform. So this is a portfolio of very robust immune assays. They are qualified and validated assays. They contain tools, monoclonal antibodies, reagents, measurements, and the main reason to apply those tools, it's a very extensive tool set, is actually to help to get to correlate of vaccine of protection for vaccines. If you were able to identify these correlates for early vaccine candidates, they would actually enable a faster clinical transition of the follow-up vaccine candidates, an important thing to keep in mind here. In terms of the question how to get to billions of doses, one thing that we all face and that we're trying to address, there's really the thought that investing in rapid protein production systems like Pichia and others can help us generate these vaccines at the scale that might be required. The other thing that we are really trying to push forward is the collaboration with development country vaccine manufacturers. And for example, the Serum Institute of India, which is already one of the world's largest vaccine manufacturers, is really an important partner in that regard, because making vaccines that are manufacturable and making them at scale and leveraging the existing capacity without having to build large investments and minimal capex is really a way to go forward here. And this is why we actually specifically designing vaccines that can be manufactured by the platforms that we prefer for distribution or that are present by these developing country vaccine partners. Coming back to this point here, there's obviously much more to talk about in terms of accelerating the research and development part. Again, here is the original timeline. Here's the very ambitious timeline of getting to vaccine candidates that are ready for implementation much faster. So there's a number of elements that one can really talk about. There are thoughts on how to increase the speed of phase 1 development. There is the thought how we need to do phase 3 and regulatory pivotal trials much more importantly. But one thing really I think that is a key element is this thought to really pull forward process development and manufacturing scale up. So these are at-risk activities, but being prepared to really make these vaccines at scale while the clinical development is ongoing is obviously risky, but it's really required to get to a portfolio of vaccines candidate quickly. So I think this is something that is being applied across the portfolio actually. This here again is just a little bit more granularity on this process development and manufacturing scale up. So obviously, you know, doing this late actually has a number of risks, because there is process manufacturing, there's process transfer activities. If these are only tackled late in the vaccine development timeline, there is a risk that things might falter. So realizing that there's a huge capacity required to make billions of doses, and that actually multiple sites for vaccine production will be needed is one thing that one can build a strategy on. Retrofitting existing sites is certainly preferred. rather than actually building new sites to do this. And again, large investments actually need to be considered to really get to manufacturing at risk in that regard. As Dick Weiler from CEPI has indicated, and we can get back to that later, is that for all of this, global cooperation is absolutely essential. So it will actually require a number of donors to come up with the costs to develop a number of vaccine candidates, which are in the billions of dollars in terms of costs to achieve this rather ambitious goal. There's a couple of slides here that are describing the XA and the COVAX pillar, but I actually will skip those because Dick Weiler addressed this so elegantly in his first talk, so I actually have nothing to add. This was enormously eloquent. And I'll just finish with this slide here with a number of really general thoughts around the COVID-19 vaccine development and delivery challenges. So the first question, and it's obviously an oversimplification, but there might be some value in going through this, is the first question is, how do we identify and how do we foster the most promising vaccine candidates in collaboration? So realizing that the initial focus was on fast, get vaccines out there fast, and actually also build them or work together with large companies with track records to get to vaccine fast. So I think this is the main characteristic of the first wave of vaccine. The second wave will likely require additional vaccine candidates, and they should really focus on vaccine platforms that are employed for scale and for distribution. I think that's an important thing to keep in mind. There's also an element in which we can try to distinguish these wave 2 vaccines from the wave 1 vaccines by vaccine design. So the current vaccines are really focusing on the spike protein as antigen. So the idea is maybe other antigens should be applied in these wave 2 vaccines. We certainly can work on improved and differentiated immunogenicity. Production advantage will be of great value. And then the thought to really initially target those vaccines on the rather difficult challenge of single dose options and improved thermal stability is going to be very important. Vaccine production, investing early, doing this in parallel before the phase 2 proof of concept is achieved, is a very valuable thought if there are sufficient funds to do that. And again, collaboration with developing country vaccine manufacturers is probably a wise thing to do, thinking towards the goal at end. Access and deliverability. So there are a number of bilateral deals that are done in high-income countries, and unfortunately, I think that is actually going to reduce the availability to some degree of these vaccines for the low and middle-income countries. So the continuous question is, what is the best infrastructure we need to meet the global needs? And I'm again referring back to my colleague Walter from CEPI here in terms of the elegant introduction that he's given to ACTA and COVAX. What we also need to keep in mind is obviously the logistical challenges that are actually associated with reaching certain risk and target groups in the geographies that we're working with, and then the vaccine-specific needs in terms of thermal stability and other quirks that some vaccine candidates might have over others, and prioritizing them in that way. I'll end the talk here with actually specifically acknowledging the colleagues in the COVID vaccine discovery team from the foundation. So this is the group of Linda Stewart, for whom I work. These are my colleagues that are pushing this forward from the discovery perspective. But obviously, there's a much larger group of individuals at the foundation that is associated with this. So I'm just summarizing them here as the COVID vaccine response group. But more importantly, it's actually the realization that there has been an enormous coming together of our collaborators, grantees globally to really then tackle this problem. So it's actually in that regard an absolutely remarkable time how dedicated people are trying to tackle this problem. So too many to list here, but obviously a great recognition and appreciation of the global collaborative spirit with which this challenge has been addressed. And with that, I thank you for your attention.
Dr. Faisal, you have muted your microphone.
Can you hear me now?
Yes, thank you.
Yes.
Thank you very much. Yeah, I was saying that you have less than 20 minutes to question. I will start with a general question. When do you think that a safe and effective COVID-19 vaccine will be available? You are mute, Dr. Marwan.
Oh, the question was to me.
Actually, for all of you.
Oh, okay, but I can't. Yeah, I think that Mr. Walder went through it and said that if it will be available, hopefully 2021 will be available. And the question is that what efficacy, and there are many questions which will remain also. That's just general answer. But Mr. Walder probably has more to add in this.
Yeah, thank you very much, Dr. Marwan. I can say a couple things. One is that there's a strong likelihood that there will be a vaccine against COVID-19 available before the end of this year, and that's saying quite a bit because we're now in November, December, so just a couple of months. But there are some, as we've discussed, there are some vaccine candidates that are fairly advanced in phase 3 trials that are showing promise. Now, whether those vaccines are available this year because they are fully licensed, that is a regulatory authority has determined that they meet all the requirements for licensure, or they're available under some type of an emergency use authorization, where they've shown sufficient safety and efficacy but haven't completed the entire process, but the demand is so great, that they will get out. It's unclear. But I would say certainly by the first quarter of 2021, many of the vaccines that we have in the COVAX facility will have advanced such that we can be confident that one or more of them will have passed through the necessary clinical trials to come into the market. We are, as was indicated, and Dr. Holger indicated this as well, that there's a lot of work that's being done in parallel to establish manufacturing capacity so that as and when those vaccines do pass through the regulatory approvals and are available to the market, they can then go out to the world in sufficient quantities. I think the reality is that in the first quarter of next year, the quantities will be relatively low. And I say relatively low, in light of the global need to have billions of doses. And we still have a target by the end of 2021 to have 2 billion doses available, for allocation on a global basis through the COVAX facility. So, I think there's reason for optimism that vaccines will soon be available. There's reason for optimism that those vaccines will begin to be allocated on a global basis. And there's reason for optimism that by the end of next year, we'll have vaccines that'll start getting us back to a more stable situation from a global health perspective. Over.
Yeah, the timeline is difficult to predict, and I think we all have access to news and we can build our own picture. But Q1 2021, I think, is realistic, just given the number of vaccine candidates that are at play. I think what's also important to realize is the point that was just made, that to make these vaccines available at a scale that will be needed globally, this will take time, unfortunately. Even with the manufacturing activities being pulled forward, I think to get to impact in terms of being able to immunize worldwide is going to start in 2021, but then is probably going to increase further in 2022. This is why I think that the field believes working on vaccine candidates that from the outset actually can be produced at larger scale is so important, right? Because at some time point, these wave 2 vaccines actually need to supplement the shortcomings that the wave 1 vaccines might have. And this is just a very important strategic element in terms of building a global vaccine timeline.
That's good, thank you. There's here a question for you, Mr. Walder. What are some of the main geopolitical challenges to COVAX with big countries like the US not joining?
No, it's a very good question. I think I think I can answer that question in two different ways. One, it would certainly be easier if the U.S. were to be a member of the COVAX facility. They bring a great deal of resource, both in terms of the research and development capacity as well as manufacturing capacity. And so, I think it's true that we would certainly welcome the U.S. to join the COVAX facility and would be able to do good things with their joining. The other thing to say is that the U.S. government, through its Operation Warp Speed, has itself invested billions of dollars in the development of vaccines against COVID-19. And many of the vaccines that they're investing in, we're investing in as well. And so, they're investing in some of the early stage research and development activities, early stage clinical trials. And we, CEPI, are investing in the same vaccines, but investing at a later stage, perhaps, or investing in scale up and scale out of manufacturing capacity so that those vaccines that are being developed with U.S. government funding can also be made available on a global basis, by transferring that technology to countries outside the United States and standing up that additional manufacturing capacity to make those same vaccines available to the world. So, and that's what we're doing, and we're in, we are in close touch, let's say, with the people from Operation Warp Speed in the United States. We follow their activities, they follow our activities, and so to conclude, to say that ideally they would be part of the COVAX process, but we are globally part of this collaboration and cooperation I described through COVAX, really taking advantage of the investments that they've made in technology and moving those technologies outside of the United States and making them available to the world.
Thank you. Sarah, here a question for you, Dr. Baker. During your presentation, you mentioned many difficulties in vaccinating people in need. Which one of them will remain as a major challenge during the pandemic or any other pandemic in the future?
It's really a good question, because I was focusing on the people in need and then the low-income countries. This pandemic is a global issue. This is beyond dispute. My major concern is me first will be always the biggest problem. I can give, of course, COVAX and trying to reach the entire world at the same time is a good plan. But I think that we are going in the end to have some challenges that have happened before earlier. It means that the well-developed countries will focus on their own nation, which is quite correct also. But when it's coming to such global pandemic, it will be great to go global, because alone you are not safe. And since we were not prepared for something like this, very contagious, I look at Nordic countries from beginning. It has created confusion and we make wrong decisions. In Nordic countries, because number of cases, they said that it was high in Sweden. Finland closed the border against Sweden. Norway closed the border against Sweden. Denmark closed the border against Sweden. And this was just totally confusion. They thought that they can control this by just closing borders. And when people going to the knee first, imagine, I mean, when they opened the border, the numbers were tripled or even more. You cannot solve the problem just thinking about your nation when it's coming to the global pandemic. So I'm really supporting COVAX, and I hope that they can reach these countries within 1 or 2 years. But I believe this will be one challenge that we are going to have it, and there will be still millions of people who will never get the vaccine.
Thank you, Doctor. Dr. Kanzler, what would you consider when you accept to vaccinate yourself or your family when the first effective COVID-19 vaccine is available?
That's a tricky question. How to answer this? So, obviously, I support vaccination, and I would get vaccinated. I believe that it might actually be a question of accessibility, because I think even in high income countries where I happen to reside, the distribution might not actually be initially for the general population. So in that regard, I tend to believe that in the end it's a safety question. It's a safety and efficacy question, right? So I tend to believe that the current vaccine development is very likely done in a very rigid and thoughtful way. I hope that there are no shortcuts being taken in that regard, and I think it actually would be a real setback for the team, for the world, that would be the case, right? So I think the primary objective is to generate safe vaccine, and then to make sure that they have efficacy, right? So I tend to believe that the environment in which such vaccines are currently generated globally will lead to safe options. So I don't have a favorite option, but I think if these vaccines have gone through the proven regulatory process, I personally would not have hesitation to receive them. So.
Thank you very much. I will go back to Mr. Wilder. I think I have here an interesting actual question. As the pandemic is spreading, how do you guarantee the efficient and equal distribution of the vaccine? Mr. Wilder, you are mute.
Sorry, thank you very much for that question. And it's really one of the more difficult ones, that we have to deal with in the COVAX facility. As I indicated during my presentation, we do have specific mechanisms that have been set up to address this question of allocation. And as I also indicated, we now have 186 countries that are participating in the COVAX facility. And we have the responsibility of ensuring that once the vaccines become available, they are allocated fairly. And again, allocated in the sense of time, that all countries of the world are in the front of the line, as it were, to get access to vaccines once they become available, and fair allocation in terms of the quantities. In the early stages, and again, as Dr. Kanzler has indicated as well, it's going to be difficult in the sense that the number of the volume of vaccine production will be relatively low. It will get greater through 2021 and into 2022, meaning that it will progressively become easier to allocate sufficient volumes of vaccines to all countries so they can address the public health concerns. And by that I mean, having an initial, perhaps relatively small allocation, but sufficient to cover frontline healthcare workers. And then another allocation that covers those that are most at risk. So people with pre-existing conditions, with lung issues that make them more susceptible perhaps to COVID would get an allocation. And at some point, there'll be sufficient volume of vaccine so that anyone who wants the vaccine can get it. But again, that's all going to have to happen progressively. Within the COVAX facility, we do have we have established allocation bodies, that will be looking at these issues on a regular and ongoing basis, and so look at what initially needs to be set up based upon the vaccine availability, based upon the readiness of countries to receive vaccines, and where they then should go first. And then that's going to be revisited on a regular basis as the number of vaccines increase, both more vaccines themselves as well as greater volume. As we gain more experience with the use of the vaccine in the field, that's going to change how one thinks about, where to allocate particular vaccines. The last thing I'd say in this respect, and this goes as well to something that Dr. Kunstler spoke about, and I spoke about it a bit as well, and this is the so-called wave 2 candidates of vaccines, is that we have this initial wave of vaccines that are under development, and I presented them in my presentation. And as we are going through that process of development, we are considering new approaches to developing vaccines, new types of vaccines, vaccines that might be more applicable in some settings than others. So this question of allocation will change as the mix of vaccines that are available changes as well, because some vaccines are showing greater efficacy for certain age cohorts. Some vaccines, may require deeper cold chain storage and transportation that may not be appropriate in some countries. And so as we see what we can do in terms of allocation to address the global health problem in this initial wave, we will do the absolute best we can to be sure that countries are not left behind in getting allocation, to be sure that we are allocating the doses as broadly and appropriately as we can to address the public health needs. And then at the same time, continue to develop vaccines, these wave 2, that we are bringing in because we're seeing, some gaps or some problems with the first wave of vaccines, and we can fix that in the second wave of vaccines. So this whole process is going to start out, I think, well, but imperfectly, because of the size and scope of the problem and the size of and the scope of vaccines available. But then it's going to get progressively better as the volume goes up, as the choices of vaccines increases over 2021 and 2022. So that was a long answer to your question, but it is a complicated problem. So thank you.
Thank you very much. Actually, the session went very quick. Lots of interesting information. At the end, I would like to thank Mr. Richard Wilder, Dr. Marwan Baker, and Dr. Holger Kanzler for their invaluable presentations and participation. Thank you very much all.
Thank you.
Thank you very well. Welcome.
Thank you.
Bye-bye.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound, where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
المملكة استضافت قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound, where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
مملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Welcome, everyone. Thank you for attending the COVID-19 vaccine conference. Before we begin, I want to thank the sponsors, the speakers, and of course the organizers as we put this meeting together. It's been quite a effort around the world to get this done, and so thanks to everyone. And this session will focus on challenges facing COVID-19 vaccine selection, distribution, and vaccine uptake. As we've heard for the past day and a half in the conferences in the meeting here, really has been a remarkable attempt at science and technology to bring vaccines to the market quickly on a global scale for this pandemic. These vaccines really vary, as you've seen, from traditional inactivated subunit vaccine technologies to emerging technologies, mostly viral vector systems, and now newly developed vaccine approaches like messenger RNA and DNA approaches. So it's really exciting to see science moving forward in vaccinology and really an application of the old, the new, and the emerging at the same time. For vaccine, there's a goal to balance potency and immunogenicity and protection. But with a worldwide vaccine effort underway, balancing these risk-benefit criteria are very difficult to achieve, and it's critical that we start to understand how to best do this. We also have to understand that there's populations that will be hard to vaccinate, as traditional vaccine work has shown us that elderly, pregnant women, immunosuppressed individuals, and young children can often be very difficult to achieve that risk-benefit factor. So, and unfortunately, there's no perfect vaccine and no one vaccine fits all. So we're also stuck with the reality that, with the pandemic, we have to balance cost, logistics, vaccine distribution, and vaccination approaches. And that becomes more complicated as we start to move forward. And this is way more complicated than our normal vaccine deployment. There's a variety of political, economical, mostly supply and demand issues, and of course, social issues to consider. And the social aspect, there's a growing resistance to vaccination, and there's a growing global distrust of science in general. So we have to overcome that. While the challenges are here, the overall goal is to provide a framework for stopping COVID-19. And I hope that we're looking forward as well as looking at the present, because as we learn how to do this properly in this outbreak, we can prepare better for any future outbreak that might occur. So it's really important for us to stay engaged in a global community, and it's really exciting to see that happening on that level. As we move forward, I'm honored to have three speakers in this session that will be talking. We're grateful to have them as part of our session. So Professor Ramesh will be presenting on efforts underway with the vaccine selection in the region. Alabata will be talking about his efforts to acquire vaccines, distribute them, and also what I'm really excited to hear about the strategies in the future is the opportunity to look forward and how can they build future capacity that we need around the world for vaccine production and even basic discovery that has to happen. And lastly, Dr. Hoots will be sharing some insights into this vaccine resistance and distrust in science and how we can overcome that and work towards a better uptake of the vaccine around the world. So just a reminder, each of the speakers will have 20 minutes to speak, and then we'll ask you, please write your questions down in the chat box, and at the conclusion of the talks, we'll have 20 to 30 minutes to talk about those with our panel members. I'd now like to introduce our first speaker, Professor Zaid Mamesh. He is an adult infectious disease doctor, and he's leading the National COVID-19 Vaccine Technical Advisory Committee and the National COVID-19 Scientific Committee under the Royal Court of the Kingdom of Saudi Arabia. This talk today will be focusing on his efforts and the team's efforts to look at how to select the best vaccines and that will fit the needs for the country and the region. So I look forward to hearing your talk.
Thank you. It's an honor and a pleasure to be with you today. And again, my thanks to the organizing committee for giving me this opportunity to speak to you today. Of course, we do know that the last emergency committee that met on the 30th of October has declared that COVID is still constitute a public health emergency of international concern. And of course, there are a lot of concerns about the rising numbers of cases over the last 2 months, consisting of the second wave. So this is data from yesterday from the WHO showing that we've had so far 47 million cases reported and more than 1,200,000 cases of death. And if you look at the graph at the left lower part of the slide, you can see that these are representing the 6 regions of WHO in different colors. and the black line indicates the mortality rates. And you can see that for our region, which is the Eastern Mediterranean region, we are in green. So our numbers are still going up, and I'll show you that in more clarity. But over the last few weeks, we can see that the United States, India, France, Italy, UK, Russian Federation, Poland, Czech Republic, Colombia, and Argentina are actually taking the lead in the numbers of cases. Well, a few months ago, we were talking about the second wave and if it's going to be true or not. And I think there was a lot of hesitation if this is going to be something that we will be seeing. A lot of publications have been done on the issue of second wave, but a lot of scientists were actually doubting that this will be something that will be happening. I can tell you it is happening, and it's happening in a very, very severe way in many regions of the world, including our own region and also the European region. If you look at the 6 WHO regions, you can see that Europe second wave is actually much bigger than the first wave. And of course, we're starting to see the increasing mortality in Europe. And also, if you look at our region in green, the Eastern Mediterranean region, you can see that our second wave is actually starting to peak, and we will be seeing more cases in the next few months in many countries of the Eastern Mediterranean region. When you look at the numbers in our region, 3 million cases to date, 80,000 deaths. In the last 24 hours, almost 31,000 cases. If you look at the countries which are suffering from the second wave, Iran is leading, followed by Jordan, Morocco, Iraq, Lebanon, and Tunisia, and many other countries in the region. This just compares on the left side the numbers of cases in the last week, and then on the right side of the slide, looking at the mortalities. And if you look at the Kingdom of Saudi Arabia, we have been fortunate that the numbers of cases have not increased significantly. So we're not seeing a second wave, and that goes to the strict measures that are being applied by the Ministry of Health and the government of KSA. And then the mortality, of course, we're still seeing the lag of the mortality from the first wave. Of course, the question that comes up, does reinfection occur, and what does that tell us about the herd immunity? If a group of individuals get infected with the virus, and then they get exposed again to the virus, some of them will get reinfected, and the other group will not get infected. And there's a lot of questions. How does that happen? Do they mount antibodies, and are these antibodies effective in preventing the disease, or are they being infected by a different mutating strain that requires a different antibody type? And that's a big question that we are concerned about. Although we don't do surveillance on everybody who's walking around in the community to identify how many of the people are infected once or twice, the numbers of cases reported to date are up to 30 or 35 cases across the globe. So I think the magnitude of the problem is not huge. I guess one of the unique things about COVID, different than MERS and other coronaviruses, is that actually infects a lot of organs in the body. Initially, it was thought that the AC2 receptors are actually the ones that seem to give access to the virus. And this is a nice diagram showing where the AC2 receptors are. And then the darker the color is, the highest the AC2 receptors are. And then when you look at the SARS virus in different organs, you can see that it actually can infect organs that does not have a high concentration of AC2 receptors. So there must be other mechanisms where this virus can actually infect different organs in the body. And these are the different organs of the body that can be infected. Almost every single organ can be affected, and the complications that we are seeing are tremendous over the last 10 months or so. So what's happening with the COVID vaccine development? The routine development of vaccine takes about between 5 to 12 years, where a vaccine starts from preclinical trials to phase 1, phase 2, phase 3, and then it goes through the licensing process, and then after licensing, safety monitoring and follow-up. This period has actually been crushed from 12 years to 12 months, and that's by the expert efforts of the different manufacturing plants across the globe, and the support that's being given by different funding agencies, whether or the non-governmental organizations like CEPI and WHO, and the coordination that is happening between the private sector and the governmental sector. Of course, the Operation Warp Speed that's happening in the US has actually pushed a lot of funding into manufacturing companies trying to develop vaccines in a very rapid way. Ideally, if we want to have a vaccine that is an ideal vaccine, it will have to have all these different requirements. And I'm sure that this is not going to be available in the near future. Every drug and every vaccine would have benefits, but also will have its limitation as far as the age groups, the different risk groups, and does it require booster? Does it work with other vaccines, and so on and so forth. We do know that if you get exposed to the virus, depending on the load of the virus, you can get infected if you're not protected through personal protective equipment. And then depending on your underlying comorbidities, whether it's being a male or being from the social minorities or being older or having other comorbidities, the chances of having mortality and morbidity is actually much, much higher. What we're hoping from the vaccine that would be effective is it either will prevent the infection from happening, or it will prevent the hospital admission, or also prevent death. So it's either preventing the infection completely or actually making it milder, and the patient will not require to go into the healthcare facility. There are concerns and problems. A lot of the manufacturing companies that are looking at outcome of vaccine, we're looking at serology to follow up and see how much neutralizing antibodies are being developed. And of course, we do have a lot of challenges with these antibodies, where we don't know how the sensitivity and specificity of different kits that are used for testing for serology, and also the timing of doing the testing and the kinetics of the antibodies within the human body. Of course, these all make an impact on the outcome. There's also a concern, like we've seen with dengue vaccine, that if we receive the vaccine, that we would have enhancement of the disease, and we get more severe disease than originally without a vaccine. There are many different platforms that are being developed. There are some known platforms that have been used for many, many years that we feel comfortable with, and there are some new technologies like the nucleic acid and viral vector vaccines, which we don't know in the long run if they're going to be completely safe, similar to the inactivated virus vaccines. Of course, RNA and DNA virus vaccine and viral vector vaccine are less established, while the inactivated attenuated virus and vector protein subunit vaccines are actually much more established. But then the new technologies is actually much easier for the manufacturing plants to produce larger quantities to cover the population of the world, which is I think 7 billion, and that will require a lot of effort in producing large quantities. When we look at the platform and the landscape of different vaccines that are being tested, we know that there are at least 10 vaccines which have reached the level 3 clinical trials, and then we have 2 vaccines which are in the second/third clinical trials. And that means that we will probably have some vaccine available by the end of this year, and most of them will be available by 2021. Of course, we have the RNA vaccines, and we have Pfizer and Moderna, which are working very hard. I'm hearing that most of the data have been collected, and I think we will have already rolling submissions have been done to regulatory agencies in the US and the UK and the European Union. And I think that will hopefully within a few weeks we will have some news about the efficacy and safety of these vaccines. We have the viral vector vaccines, which include the Chinese products, the CanSino, we have the Gamaleya Russian product, we have the Janssen pharmaceutical product, and of course we have the AstraZeneca, which I think is in late clinical phase 3 trials. And again, we expect to have a vaccine by the end of 2020, and if not, in the first quarter of 2021. Of course, inactivated vaccines, we don't have too many of them being produced, but there are at least 3 of them being produced by Chinese companies. And of course, the protein subunit vaccines, Novavax and Sanofi Pasteur. Of course, we do have some issues with these vaccines. They are being trialed in patients who are 18 to 61 years of age. Some of these companies have actually extended that to include older age, but we don't have a lot of the vaccines being trialed in older age. Why do we worry about people who are older? Because we know that the immune system will be much weaker. They will not have as much T cells and B cells, and also their immune response will be dubbed and reduced. So they won't be able to mount a good response to a vaccine. So this is an area where it needs to have a lot of testing. The other area that we're concerned about is, of course, children, because none of the trials have actually included any children in their clinical trials, and also the obese individuals. We think that the obese people are having increased morbidity and mortality, and we think they will require adjustments in the vaccination program. In Saudi Arabia, when we looked at the different vaccine platforms, we were looking at the speed of vaccine availability, the site of vaccine manufacturing. We're looking for manufacturing sites and companies that have good experience in vaccine development, and also the platform safety. So, the new technologies tend to, we have to wait and see in large numbers of individuals if the long-term safety will continue to be supported, and also the logistic needed for vaccine transport administration. And we know that the mRNA vaccines will require -70 or -20 freezers at the site of administration. Of course, populations that will be prioritized for giving the vaccine early on is people who are at highest risk, which is including the healthcare workers and people who are on the front line dealing with the public, and then elderly who are more than 65, and then people who are younger than 65 who have comorbidities. This will not we will not be able to cover the whole population of every country with 7 billion doses, because we know that all the manufacturer plant capacities across the globe will not be more than 2 billion doses by the end of 2021. So I think we will have to target the people that will be at higher risk and where the benefits will be the highest. Of course, we know about the ACT Accelerator, which is a program developed by the WHO in collaboration with CIPI and Gavi. And basically, the whole idea of this program is to develop the supplies needed for COVID, whether it's a vaccine or drugs or diagnostics, and to be able to distribute them equally to all countries across the globe. We do know that the COVAX facility, and which is in charge of the vaccine development, is doing very well. They actually have a few vaccines in their platform, and then they are working with the countries to ensure good distribution of the vaccine to countries across the globe. So basically, the ACT Accelerator is planning to develop 2 billion doses of vaccine by the end of 2021. Develop 245 million courses of treatment by mid 2021, and 500 million tests by mid 2021. And I think they have been doing very well with 9 vaccine candidates already in portfolio. They have 1 new treatment secured in low and middle income countries, which is the corticosteroids, and the diagnostics are moving very fast, and allocation with equity is being done in a very, very rapid way. I think the question is, once all these vaccines become available, would people actually take them? And this is a nice study which looked at multiple countries across the globe in different regions of the world, and the actual acceptance of the vaccine ranges between 50% in countries like Russia and Poland to up to 89 or 90% in China and Brazil. So in our region, we're still doing the work to find out the acceptance of the vaccine once the vaccine becomes available. Of course, we are sometimes challenged by not knowing a lot about what's happening with side effects on the vaccine side. We know that AstraZeneca had to stop its studies once, and Janssen has stopped the studies. And I think the reason why a lot of information is not being made available is to protect the vaccine and to protect the material, the data that the companies are actually securing, to ensure that the data is being reviewed by an independent board to know its efficacy and safety. I think one last thing before we close, I think to stop the next pandemic, we need to unravel the origins of COVID-19. We are now 11 months into the pandemic, and we still don't know what the source is. This is a very nice opinion piece written in PNAS yesterday, and I thought it would be worthwhile sharing that with you. So in conclusion, we have to balance the safety versus speed, and we know that old and known is much better than new and unknown. Operation Warp Speed is targeting genetic vaccines and virus vector vaccines, which are new emerging technology. Although 30,000 patients are being enrolled in phase 3 trials, we cannot tell the long-term safety until at least 20 million doses are being given. So we're being extra cautious, and of course, we're hoping that all the regulatory agencies will have a system of monitoring the vaccine post-licensure. Inactivated weakened virus vaccine and protein-based vaccine are probably the safest and most widely marketed that we know of. More than one vaccine platform should be targeted for purchase, and that's what's happening in many of the countries. We have to ensure active membership of COVAX facility, and that's happening with KSA. We should procure small quantities from different companies so we have a higher chance of one vaccine being successful. Be aware of rising vaccine distrust and target the highest risk group early on, healthcare workers and frontline workers. And of course, pregnant women and children should not be given the vaccine early on until more data becomes available. I will stop here and thank you for your attention.
Thank you, Professor Mamesh. That's a wonderful overview and really helpful in understanding the selection process and how things are moving forward. As we move to our second speaker, Dr. Yasser Al-Abada, he's a CEO of Pseudo Air Pharma company based in Kingdom, Saudi Arabia. I'm looking really forward to his talk. He's going to talk about the long-term investments that have been made to ensure the healthcare system in the Middle East and North Africa are taken care of, and also working on how to look into the future past COVID-19 and how those investments can better prepare the region as we move forward. So thank you.
Thank you very much for this introduction, and I would like to thank the organizing committee to select our company to present something which is different than a scientific. We will go to the business more than scientific, which is we started to study how we can invest in biopharma. That's why I will not just focus on vaccines in this presentation, but I will speak in general about all biopharma business. Next slide, please. Is the control with me or? Yeah. First of all, we'll speak about the Saudi Arabian pharma market, which is one of the largest spender on healthcare across the Middle East and North Africa. For the SAGIA, which is one of the organizations for investment, they said in 2018, government budget allocate almost $40 billion to health and social development, and which is the growth of this market are almost 5.6%. The spending on patent medications in Saudi Arabia more than $4 billion. When we see the overall cumulative data that we see from 2017 until 2021, almost we have $42 billion in this market, especially on the pharma. The generic almost 2.3 billion. The biosimilar size and all of these job creations, one of the governmental agency called the National Industrial Development and Logistics Program, NIDLIP, they put a pharma in Saudi Arabia, one of the priority for the industrial development. And they said that this market are increasingly almost 8% in Saudi Arabia and the kingdom, almost the total market focus on the imported medications, which is almost 80% are imported and 20% are locally produced. If you go to the vaccine here, only 1%, which is almost secondary packaging that we have it, but 99% in Saudi Arabia for assembled vaccines that imported, not manufactured locally. So that's why on the NIDLIP program, they said we need shift to more advanced product to become a leading and innovative country in MENA region, especially in the pharmaceutical and biopharmaceutical industry. Biopharmaceutical is really a term which is, we have a little bit of mixing of understanding, but one of the oldest biopharmaceutical medications, which is the vaccines, which is considered as a bio product because, as a definition, the bio pharmaceutical, which is drug produced by biological source by using the biotechnology. And we remember that Edward Jenner, which is he developed the first vaccine in 1797, which almost coming from cowpox to try to develop the smallpox vaccines. That's mean the biotechnology, biopharma, it's very old technology, but nowadays are more increased because the current medications, especially for chronic medications, for diabetic or antihypertensive, usually the medications we have 25 to 30% of the population having the effect, like reducing blood pressure. Certain percentage they have no effect. Certain patients they have adverse drug reactions. That's why you have to stop the medications. But the future, what we call it, personalized medications and diagnosis, that from the DNA that from the patients that we can give for each patient the special medications that it's really having the effect directly. If you see here the biopharma versus the conventional pharma, it's moving that the biopharma, for example, in 2010, it was almost 35% by value. But if you we expected in 2024 that the biopharma almost will have 50% of the market, that the value of these medications, it will be most of the budget going to the biological medications. If you see the biopharma target therapies areas, that we have a lot of diseases now. We have these kind of studies and medications that under development. So if you see for infectious disease, almost 176. This is before COVID-19. If you see like cancer is almost more than 336 medications and other specialties that the biopharma is coming and promising in order to have more target and to cure the diseases. If you see on the COVID-19, the biopharmaceutical, we have almost in September 2020, more than 590 drug development programs are in planning stages, and the FDA alone has reviewed more than 310 trials on the end of last August 2020. That's mean a lot of these medications, especially vaccine and non-vaccine, are targeted for COVID-19. The movement of these medications are increased because of the demand caused by COVID-19. It's really very sophisticated techniques, especially when we go, if we want to localize on the larger scale biotechnology manufacturing facility, we require from 200 to 500 million US dollars. And also you need 4 to 5 years to build. If we want to have it like small molecules, it's almost 30 million to 100 million. That's mean the investment here, it's really big, but it's we have very rapid growth and increase the importance of this industry, especially for the new challenges that we have it, and also we have an opportunity like COVID-19. Just our brief discussion about two very important areas that we like to have it and invest on it in Saudi Arabia, which is two new therapy, which is advanced therapy, which is the cell therapy and the gene therapy. This is very expensive therapies, which is we have one product are registered in Saudi FDA recently for Novartis is 1,600,000 Saudi riyal, which is 435,000 US dollar for one medication used for certain lymphoma. And also the gene therapy. And we have also almost 263 products are now in phase 1. We have 53, and phase 2 we have 1. That's mean these medications will come in very, very, very fast, and all of these medications is very expensive. and it's the problem who will be these kind of very expensive medications. The same story for the gene therapy. One of the products are used for inherited retinal diseases is almost 1,700,000 Saudi riyal. That means very, very expensive medication for each patient. This is the companies, if you see the landscape of the biotechnology, which include the COVID-19 companies, we have more than 900 different companies that they are really working in worldwide in gene and cell therapies and tissue reengineering. If you see the North America is the most important area, which is more than 480 companies. They are starting in these technologies, and I think in COVID-19 these numbers are increasing. If you see in our region, Europe and Israel, they have more than 240, but in our region, especially in the Middle East and Arabic countries, we have no companies are really investing in these new therapeutic technology for gene therapies, for development of vaccines, and hopefully we trying to be first company. These kind of technology is really having a lot of challenges, especially on the logistic and manufacturing, and all of these steps are very complicated. It's not simple issues for each individuals to have, because some patients we collect from them the sample, and after that the transport logistics, especially in Saudi Arabia, and manufacturing is really a big challenge. Here in Saudi Arabia, we have a lot of medication will be above the patent, especially the biologic. We have the most expensive biologic medications. We have 10 products, which almost revenues currently total more than $81 billion for this 10 products. And these 10 products are really very important products for the biologic, that it's really, some of it are used in COVID-19 treatment, but this kind of product will be expired, and hopefully we will invest in the biosimilar of these products. One of the strategy that most of big pharma they are using, which is merger and acquisition. If you see in the blue color, certain companies like Gilead, for example, they acquires Kita, which is almost $12 billion. If you see another company like Novartis, they acquire Avixia, which is almost $8 billion. And this is a merger and acquisition, one of the areas that it's very important investment in the biologic, especially on vaccine development. This is very important issues for vaccines or biopharma in general. The platform that it's really very important to have the ecosystem, which is really very complicated, because usually to start with academia, which is research, and after that we have a support, we have a capital providers, regulatories. All of these should talk with each other. It's not the companies. The company is one sector, which is other important sectors are very important to each one of these players to talk with each other in order to provide, for example, a vaccine for COVID-19. You have a lot of talk. That's why if you see, as Dr. Ziad, he mentioned, we have an accelerated approvals, because there is a lot of organizations from academia, from capital providers, from regulatory, from CDMOs. All of these help the companies in order to develop the vaccines as soon as possible. This important platform, which is we don't have it here in Saudi Arabia, which is that's why we need to focus on that areas, because it's not for the new biopharma. It's not only the companies who playing the important roles. We have another player that is they have more important rules in each step of the drug approval, because the biotechnology is really complicated. I remember one of the studies done by biotech organizations to see the likelihood of the approval of the biologic for the last 10 years, one medication, 10% of these medications are approved and 90% fail from the phase 1 or phase 2 clinical studies. That means the investment here is a very risky investment in biology. One of very important issues that we have in Kingdom of Saudi Arabia, which is we have high burden of genetic diseases both in severe inherited diseases, which is show up early in life, which is 80% of our burden in Saudi Arabia. They have inherited diseases, and also some of the diseases show up later in life, ultimately impact over 20% of our population, like diabetes and other. And we'll have really a national cost is really cost us the country more than 30 billion annually for healthcare because of certain diseases. One of very important step that the government and here Prince Hamad bin Salman announced the program, which is Saudi Genome, and they have samples for over more than 50,000 Saudi citizens, and they discovered a lot of genetic problems. One of, I think they have more than 1,200 different diseases caused by genetics in Saudi Arabia. And all of those diseases can be cured by prevention and by cell and gene therapy, especially gene therapy that we can use it in Saudi Arabia because a lot of marriages are families, and we have a lot of big issues like sickle cell anemia that we have it in Saudi Arabia, and we can solve it by prevention and by treatment. What is the limitations in the challenges of localizing biopharma, especially on the vaccines issues? We have limited national clinical research capabilities, and also the regulatory framework, we have certain limitation. Also the financing and funding resources like grants, also we have certain limitation in that. The skilled staff and manpower also one of the limitations which we discovered when we decided to invest in this area. And one of the issues, small local and regional market. We don't have big market because of the population that we have, and no clear purchasing reference policy for locally produced medications. We are, as a company, we put our vision in 2014 that we want to fulfill the unmet need of pharmaceutical sectors. And that's why we try to localize an advanced biologic industry in healthcare. We don't produce any general medications. We concentrate on cancer therapy, respiratory vaccines, and biosimilars. And all of these facilities, we started with the first facility. This is our complex, which is the phase 1 already started, and phase 2 and phase 3 and 4 it undergoing. This is our model of backward integration in order to transfer the technology to our staff, especially because most of our staff are Saudis, for Saudi, because we want to improve the human resources in this kind of technology. That's why we put a backward integration to be easy for us to transfer the technology. We have focused on and committed to the quality from the international standard, especially on the efficacy and safety across all our operation. And we have to deliver and to be available as the challenge that we have it in COVID-19. It's not all the countries they can have this vaccine available for them. Affordability, we try to do bring the expensive medications within the reach of our patients. What we did here, we actually sign an agreement through our partners to localize vaccines. We have Sinovac, we signed with them for hepatitis A vaccine, and we're trying also to introduce them to Saudi Arabia and COVID-19, one of very important Chinese company. Serum Institute for General Vaccinations and SK Bioscience, all of these are already we signed this year with them in order to transfer the technology to localize 11 vaccines in Saudi market. We signed a big pharma company which is producing biopharma, especially on the biosimilars, and they will transfer all the technology. We signed MOUs with the universities, hospital, and clinical research like KeyMark, which is one of very important player. They will help us to localize all of these important technologies. And lastly, we have a different discussion with the US and Chinese company to start the CAR-T cell with either GMP facility or closed system. And this is inshallah next year we will start maybe to build this facility. What is our recommendation in conclusion? I think when we go to invest in biologic medications, especially vaccines development, it's not a private sector capability. We, from our experience for the last 4 years that we're trying to search and invest in this area, we have to have a company owned by the government to regulate the investment in biological, because certain biological medications is very risky to invest for the investor to take this risk. The government should own this company with the 50%, with the 30% of local manufacturer to be transferred here and localized, and 20% for universities and governmental institute also to be part of this company in order to draw the direction roadmap for these kind of investment. Also, we recommend to buy shares in startup companies in biotechnology, which is very important companies in Series B and C business to establish a base. And later on, if there is any success in registration, they can also transfer it in Saudi Arabia. Investing and managing in balanced R&D portfolio. For the long-term investment, gradually we have to build the R&D capabilities in biologic through the clinical trials facility. And we saw here in COVID-19 pandemic that the need of this R&D, all the countries are looking for a big pharma to solve their problems. If they have a capability to have certain research, especially with the Saudi genome, we have a lot of information about the Saudi population genetic issues that can help us to build very good bio medications, either vaccines or treatment medication for our region, especially on the MENA region. We have a lot of needs of these kind of biologics. Thank you very much for your time. This is a brief about investing in biological medication, include vaccination in Saudi Arabia, and our experience as a company, a local company, in investing in this area. Thank you very much.
Thank you, Dr. Al-Arabi. Exciting future for the health of the MENA region as a whole, and it's great to see the investment that's being made. As we move to our next speaker is Dr. Peter Holtz. He's from Baylor College of Medicine. He's a widely respected expert on vaccine development. If you live in the U.S., he's on the TV and radio quite often, and podcast. Thank you for being on the call today, for the talk today. And really, his talk is really about how we dispel the myths and misconceptions about vaccines and science, and how to change the anti-science disinformation approach that we're seeing in the world right now. And of course, a long-term benefit to public health that could be achieved through vaccination that has been lost on current generations on how they're against science. Dr. Holtz?
Well, thank you very much for the invitation. Our group will actually have two presentations, one by myself and the other by my science co-partner, Dr. Mary Elena Bottazzi, and she's going to discuss our low-cost recombinant COVID-19 vaccine that's now being scaled in India by Biological E. They're preparing 1 billion doses of a vaccine that's similar technology, a yeast-derived recombinant protein technology similar to hepatitis B, but for the receptor binding domain. that looks very promising in terms of virus neutralizing antibodies and protection in laboratory animals. And we're looking now at licensing that to other countries as well. We're in discussions with about 5 countries, including 2 in the Middle East, and hopefully this will make a contribution to COVID-19 in the Middle East. But today, this morning, for here in the U.S., I'm going to talk about wear a different hat, in that in addition to being a vaccine scientist developing coronavirus vaccines and other vaccines, I've also been working to fight this terrible, what was an anti-vaccine movement, but has now turned into a full-fledged anti-science movement. And it may not be so bad in Saudi Arabia, but globally it's become a very powerful force and is really worrying me. So I got involved in this whole area because I have 4 adult kids, including Rachel, who has autism. And I wrote this book called Vaccines Did Not Cause Rachel's Autism. And that got me on a parallel path of going up against anti-vaccines and anti-science movements. And today I want to talk about how what was a focused anti-vaccine, anti-science movement in Texas eventually became national and now global. And now I have a new book on the left called Preventing the Next Pandemic, Vaccine Diplomacy in a Time of Anti-Science. And I talk quite a bit about my time as U.S. Science Envoy in the Obama administration, when I made 5 visits to the Kingdom of Saudi Arabia, which I loved. I loved my time in Saudi Arabia, and I see some familiar names on the list, and it's good to see old friends and colleagues. And it's an honor to be back. I just wish it wasn't virtual. I wish I could be there in person, but inshallah, maybe as this apocalyptic pandemic ends, I can make another visit. One of the problems the way I'll start this is to say that we already have a problem in the U.S. that up to half of Americans will refuse COVID-19 vaccines, even if they're made available. And a lot of this it has to do with the messaging or lack of messaging coming out of Operation Warp Speed, which is the U.S. vaccine program to accelerate COVID-19 vaccines. And I want to really address how this evolved to the point where we may be in a situation in the U.S., even if vaccines are made available, they won't be used because of suspicion and distrust of the government and distrust of the vaccine process. And the point is, a lot of this was predicted and predictable based on the evolution or de-evolution of the anti-vaccine movement. And it really started more than 20 years ago with this publication in a fake paper by Andrew Wakefield and his colleagues, claiming that the MMR vaccine, the measles, mumps, rubella vaccine, especially the measles component, had the ability to replicate in the colon and the gut of children, and then that led to autism, or what they then called back then pervasive developmental disorder, or PDD, but basically it was autism. And it took Brian Deere, a journalist for the Times of London, who ultimately wrote a series of articles in the BMJ, the British Medical Journal, to reveal a pretty elaborate fraud scheme that Wakefield and others were involved with. And the paper was retracted by The Lancet, but not until 2010. So between 1998 and 2010, that 12-year period helped really give rise to an anti-vaccine movement, to the point where last year, before COVID, the World Health Organization declared what they call vaccine hesitancy one of the top 10 global health threats for 2019, because through declines in vaccine coverage, you had 80,000 measles cases in Europe in 2018, and 90,000, some say even 100,000 measles cases in 2019. And so measles had basically come back to Europe because of declines in vaccine coverage. And it was not only in Europe, it really started off to take off in the United States, and to the point where we had measles in several hotspots in the U.S., where we had found that large numbers of kids were being denied access to vaccines. So last year, like in Europe, measles returned to the United States some 20 years after its elimination. So this had reached a critical mass point where vaccine hesitancy had reached the point where now we were seeing the return of epidemic infections led first by measles, and that's usually the first one you see because of its high transmissibility and its high reproductive number. And this is the situation that we faced here in Texas, where I live and work. We are now up to 72,000 kids being denied access to their vaccines in the state of Texas. And we also in Texas have a lot of homeschooled kids, up to 300,000 homeschooled kids in the state of Texas. We have no idea of the percentage of those not getting vaccinated. So it's likely we have well over 100,000 kids not getting their vaccines in the state of Texas. And it's particularly concentrated in the Austin, Texas area, although we see it quite a bit up around the Dallas area, in Plano and Denton as well, in North Texas. And I know a lot of Saudis visit Texas quite a bit because of our links with oil and gas. And so Texas has become quite a concerning place. And what happened in Texas after the Wakefield paper, it took this very odd, strange turn to the extreme political right in 2015, coinciding with the election of the presidential election in 2015, 2016. And it was under this banner of what was called health freedom. That was kind of the term they used, medical freedom or health freedom, saying the government can't tell us what to do with our kids. And so we saw this steep increase in the number of kids not getting their vaccines. And now what's happened in this time of COVID-19 is those same health freedom groups and medical freedom groups are not only protesting against vaccines, they're protesting against social distancing and contact tracing and masks. And this is one of the reasons why we had that massive surge of COVID-19 during the summer was because people were defiant and wearing masks and refusing to engage in any kind of social distancing behavior. So what's happened now in that environment, you have Operation Warp Speed. And unfortunately, the language being used by that term, Operation Warp Speed, has helped fuel the anti-vaccine movement. So among the things that the anti-vaccine movement claims is that vaccines are rushed, or they're not adequately tested for safety, or they claim there's a conspiratorial relationship between the pharma companies and the U.S. government. And I mentioned health freedom. And of course, they still claim that vaccines cause autism and other chronic illnesses. And the problem that we've had with Operation Warp Speed has been that there's been no communication strategy around Operation Warp Speed. I think the scientific program is going pretty well. I think the scientific rigor is excellent. I think the integrity of the clinical trials is strong, but there is no communication strategy with Operation Warp Speed. It's being left to the pharma CEOs, and the pharma CEOs are not doing a good job in their public relations messaging. You had with Moderna the allegations of inside stock trading, and then with AstraZeneca Oxford, you had the CEO having leaked a call with JP Morgan investors about the pauses in the trial because of potential safety concerns, and this is how the American people found out about it, and it's not going well. So I think it's going to be urgently important that we put together a communications team and strategy for Operation Warp Speed. Otherwise, we're going to continue to have the vacuum filled by this very aggressive anti-vaccine health freedom movement. Now, the other thing they claim is vaccines cause autism or other chronic diseases, and I've gotten involved with that because, as I mentioned, I'm a vaccine scientist, but also the parent of an adult daughter with autism, and I wrote this book a couple of years back called Vaccines That Not Cause Rachel's Autism. That's Rachel over here. And it goes into depth explaining the evidence that there's no link between the MMR vaccine and autism. There's no link between thimerosal in vaccines, as the anti-vaccine lobby claims, or spacing vaccines too close together, or aluminum in vaccines. And then from there, I go into some detail explaining what autism is and how it begins in early fetal brain development. This is a paper that came out earlier this year from the Broad Institute at Harvard MIT, doing large exome sequencing, and where 102 genes have been now implicated in autism spectrum disorder. All but one are expressed in the cortex, expressed in early brain development. Most are expressed in enriched and excitatory inhibitory neuronal lineages. A lot of them are in the neuronal cytoskeleton. And one of the things that we did, because Baylor Genetics is very strong, is we did whole exome sequencing on Rachel and my wife and I. And one of those genes is a new autism gene, not in the 102, but it does encode a neuronal cytoskeleton gene, a non-red cell spectrin and neuronal spectrum gene, which is now being written up in a series for publication. And the other interesting piece to this, reinforcing the genetic basis of autism, you know, the anti-vaccine lobby likes to claim that the highest rates of autism are in the U.S. and in Europe due to the fact that we vaccinate. In fact, it's not the case. It's more very much the Middle East and North Africa, where we just heard that there's a high rate of genetic conditions, and this goes along with that. So what we're seeing, to finish up, is that we're seeing this this globalization now of the anti-vaccine, anti-science movement. You have the Russian government through Russia Today, their media communications flooding the internet with anti-science, anti-vaccine messages, and you have the leaders of the anti-vaccine movement in the U.S. now marching. There were marches staged in Trafalgar Square in London, in Paris, in Berlin, Germany, against vaccines and contact tracing and masks. And this is one of the leaders of the anti-vaccine movement in the U.S., Robert F. Kennedy, at a rally in Berlin, Germany, which CBS News and others reported is supported by QAnon and even neo-Nazi groups. So there's a very dark turn that's happened with this, and we're hoping to counteract it. We have this problem of vaccine nationalism already, where now we talk about the Chinese vaccine, the British vaccine, the Russian vaccine. We never used to speak that way about it, but part of this is we've tied vaccine development to nationalism. And I'm worried about this because I think you're starting to see countries, especially in Latin America and Africa, make one-off deals with the Russians, the Chinese, and others. And we've always operated on these principles of vaccine diplomacy, international cooperation between countries. Like when Albert Sabin developed the oral polio vaccine, he did it jointly with the Soviets at the height of the Cold War, two countries putting aside their ideologies. And that was my role as U.S. science envoy, to build vaccine development cooperation between countries in the Middle East and North Africa, including Saudi, with the U.S. government. And I think we need to continue in that important direction. So to finish, I'll just say we are starting to see a bit of an unraveling of the global governance of vaccines, and it's happening in part because of vaccine nationalism, but also anti-science and things that we don't ordinarily think about as biomedical scientists, things like urbanization, political instability, or even climate change. And on that basis, we've just created a new vaccine risk index to combine all of those to identify the areas where we'll see vaccine preventable diseases return. So I see us involved in this epic struggle. We're making great progress in our ability to develop new vaccines and biologics, including monoclonal antibodies, but we're being opposed by this new array of forces that most of us were never trained to think about, like anti-science, poverty, climate change, war, political collapse, urbanization. And I think we're going to have to really be able to address this in a meaningful way. And I think this will be very important for the upcoming G20 summit in Saudi Arabia to really look at this, because COVID-19 turns out just to be a culminating event of a trend that's been going on for several years. So with that, I'll stop, and I'll proceed however you'd like to in terms of next steps.
Thank you, Dr. Hoods. Your message reminds us that we're not done once the vaccine reaches the patient. And it's a great point about the global cooperation that's needed in a public health event like this, and avoiding the ideology and really working together for vaccinations and approaches that are accepted by the world, not just by a nation. That's wonderful conversation to have. So I'd like to start off with just a general question for all the panelists. If you can turn your screens back on real quick. The question is really, how do we learn to speak the same languages between ourselves, business, government, science, and the public? I think this is, we have this is us versus them with the public, but it really goes much larger than that. It goes into the realm of, how do we talk, from a business perspective when we're a scientist, and how does a scientist, talk to the government? And of course, how do we make the public, who generally in most organizations or most countries is responsible for funding the science that drives this? So how do we get that global, expectation between the different individuals? How do we manage that? And there's no order. Whoever wants to speak first. Okay, Peter.
Yeah, I think that's a great question. It's a very profoundly important question. When I was getting my medical degree and PhD in New York back in the 1980s, and I'm sure like many of my colleagues here, the message was you're not supposed to talk to the public directly, or you're not supposed to talk to journalists. That was seen as self-promotion or grandstanding, and all of these ideas that just scientists can only speak to each other was put in place before something called the internet came along, and before something called social media came along, and it's created a vacuum. And I think our inability or unwillingness to talk directly to the public as scientists has created a vacuum that's enabled anti-science movements to flourish. And I've been writing about this and making the recommendation that among the things we have to think about is to build in to our doctoral and postdoctoral training science communication and public engagement. I had to learn it by trial and error, probably more error than trial, but I think this is going to have to be a new approach, and training it's not for everyone. Not every scientist wants to do this, or physician, but building in those opportunities, because I think one of the things that's happened with COVID-19, at least in the US, is people do want to hear directly from scientists. They don't want to see everything filtered through politicians and elected leaders. And I think if we did that, it could be extremely effective.
That's wonderful comments there, and I think Dr. Alabata, I'm really excited to hear your kind of point of view on this too, because from the business side, that's where the work gets done most of the time, is where there's a exchange of funds. So.
Yeah, I think during this pandemic issues that here is not beyond the business. I think the cooperation, and we have to talk with each other from each aspect, because we are facing a big international problem. All of us are being affected, our families, that we have to cooperate beyond the business. And that's why when in my presentation, I said in this vaccine development and all biotechnology or biopharma issues, it has to be from the government, from the private sector, from institute, from the scientists, sit together and put a plan together in order to rapidly, this COVID-19 is, I think it's a starting point, which is we don't know which later on we have a different pathogen that we will have, and we'll have different issues that affecting our life. We have to be aligned together and talk together, and that's why I put it in the ecosystem for this kind of technology. All of us should really have a common language to talk, from the scientists and from the academia and to the government, even the investors, the companies. All of us should be united against this pandemic problem.
wonderful point about the ecosystem. I think that's a kind of a key point to that. Putting us all together in a common place is helpful. Dr. Mamesh, do you have any comments on that, on this first opening question?
Nothing to add to what Peter has said. I think he put it very eloquently in his response.
Okay, so another question that this is one of my personal question for the team is the group as a whole. The role of comorbidity increases the risk of disease. We've learned that around the world, and there probably will be some interesting data that, from an epidemiology point of view, we'll see that healthy communities will have done better against COVID-19. And so as we move forward, one of the things I came up with just recently with a colleague is that type 1 diabetes in the U.S. Is a high risk condition for complications, yet type 2 is not. So even managing a disease, so type 1 diabetes, typically are patients better at managing their care and taking care of themselves than type 2 in general. So clearly, again, healthy society can have a benefit against emerging disease. So how do we manage that kind of healthcare dilemma and paradox of having healthy people, but at the same time being prepared for an outbreak?
I'll just say a word in that, this link between diabetes and hypertension and COVID-19 is not totally a surprise, because we see this with other conditions. So for instance, in India now, the reason why so many are getting very sick from dengue is, we always thought of dengue as a pediatric infection, and it still is, but we're seeing lots of high morbidity and even mortality from dengue in people with underlying diabetes and hypertension. Here in Texas, especially in South Texas, where diabetes is also very high, severe morbidity and mortality between diabetes and tuberculosis. So we are seeing more and more this interface between emerging infections and non-communicable or metabolic diseases and conditions. And both are growing. So I think we'll see more of this kind of syndemic or twin epidemic of infectious diseases and metabolic conditions and non-communicable diseases. That's going to be the new norm in the 21st century. And it's interesting, we don't really quite understand the mechanisms linked to that. We don't really why do people with obesity or diabetes or hypertension do so terribly with COVID-19? There's some hypotheses out there, but I don't think we really understand that well. So doing more research on this, I think, is going to be extremely important. And also getting away from this paradigm of saying, well, forget about infectious diseases, now we have to worry about non-communicable diseases. Well, COVID-19 threw that out the window anyway, but the point is the two go together. It's not either or. There is an unfortunate synergy there. I think you're muted, Gene.
Sorry. Any other comments from the other panelists regarding that question?
No, just to say that I think the other issue that we're seeing with this pandemic is the excess mortality from unknown causes. We know that people who are having chronic diseases are not actually going to the healthcare facility to seek care. So the elderly, the people who have severe comorbidities, the number of visits to the ER for STEMI and non-STEMI and other complaints are being reduced significantly, and I think this will lead to increased mortality at home. So this is something that also need to be looked at very carefully over the next few months.
Yeah, I think one of the messages from COVID, when it first emerged out of Wuhan, we all thought of it as just another version of SARS-1, and that it primarily is a respiratory illness. And it is a respiratory illness, but it's probably more vascular or cardiovascular illness, and the thrombotic events, the pulmonary emboli, the strokes, the coronary artery thrombosis, and the heart attacks. This is what worries me about getting COVID-19, either for myself or my family, all the thromboembolic phenomena, which again, we don't really have a good understanding about. But I think the other thing to keep in mind is that COVID-19 will not be our last coronavirus pandemic, right? We've had SARS in 2003. We had MERS coming out of the Arabian Peninsula in 2012. Now we have COVID-19. Others will surely follow. There will be COVID-22 and COVID-28, and we can count on that. So we're starting to now look at what will be needed for a universal coronavirus vaccine, whether it's combining all of our antigens for the different coronaviruses to make a polyvalent vaccine or trying to come up with common universal sequences. This is going to have to become a priority as well. Just like universal flu vaccine, we can count on the fact that we'll see coronaviruses now every few years.
Good points. Moving to the kind of a comment from the chat box we have. Now that we're awaiting the successful vaccine and safe, approach that we will have available to us, how are we preparing for the upcoming vaccine challenges, rolling out the vaccines and challenging education? How do we educate the public? How do we get the message across that this is valuable? And how do we simplify the widespread logic and concept about vaccines being used for wrong reasons and that won't offer value to the average person without a scientific background?
I think that's going to be a tough job. I read recently in the Annals of Internal Medicine, a nice report on what the physician should be telling their patients when they see patients who are asking about the vaccine. And I think it's a huge responsibility and a huge burden because there are so many things that we know, but there's also a lot that we do not know. And I think as long as you're honest and transparent to your patients, I think that's the best that you can do. I think maybe in my simple mind, I think the best thing that you can do to your patient is try to suppress wrong information. So if they come to you with information that is not correct and collected from the YouTubes or from the social media, then you can actually negate this information and give them the accurate ones. But then you will have some challenging patients who can ask you tough questions, and it depends on how this is explained by the physician to the patient that will make them, decide to take the vaccine or not.
One of the challenges that we have in this coronavirus vaccine is that the time from starting preclinical studies until the phase 3, which is a very short time. Sometimes you don't have a full information that you have it. Usually, if you want to develop any biologic, especially on vaccines, you need from 7 to 12 years until you have a lot of data, a lot of patients, the efficacy, the safety, the preventability of these medications are really there that can help you to explain to your patients. Our challenge here, we have really very limited information, even as Dr. Ziad, some of the companies that is not sharing a lot of data because of the protecting their rights and all of these. That's why healthcare providers, they have really very limited information that it's really can answer all the tough questions that the patients can ask them when they visit them.
Good point.
I think the other piece to that is, again, there's a few moving parts to that. One is the fact that there's going to be require an unprecedented amount of public health communication around these vaccines that we've never had to do at this level. I mean, you saw the list of the different vaccines that even Saudi alone will be using, possibly one from Serum Institute, Sinovac, maybe others, maybe ours. They're going to have different levels of protection. Some may reduce severity of illness. Others will actually stop the infection, but we won't know which. And if they actually stop the infection, we're going to need a certain percentage of the population to take the vaccine to achieve herd immunity, or they may be vaccines that don't stop the infection but reduce severity of illness, in which case we're still going to need masks and contact tracing and everything else. We have no idea of the durability of protection. They're going to need boosters. Do they get boosted with the same vaccine or a different vaccine? All of this needs to be communicated. And one of the things that I've been saying, the question I always get asked is, hey, Dr. Hotez, which vaccine are you waiting for? And I say, I'm not waiting. I'm very uncomfortable going through this winter without virus neutralizing antibodies in my system. I will take any vaccine that's offered to me that's approved, and then even if it's not the best vaccine, and get boosted later potentially with a better vaccine. But again, none of that is really being messaged. So it's not certainly totally not happening in the U.S. government. I don't know about Saudi, but creating a pretty detailed communication strategy around the vaccine with trusted government scientists who are out there on a regular basis communicating with the public is going to be really essential. And so I think that's a big piece of it. And and a lot of people accept what I say when I talk about that part, because one of the things that I sometimes depart with my colleagues is then they say, well, we've got to fine tune the message. And I say, well, if you only worry about fine tuning the message, remember it's still a message in a bottle in the Atlantic Ocean or the Indian Ocean, because we've got this very aggressive anti-vaccine lobby out there that dominates the internet. There are 480 fake anti-vaccine websites out there, all revved up on social media. If you go to the Amazon.com site for books on vaccinations, they're all fake, and everyone's a fake anti-vaccine book, claiming vaccines cause autism, that we don't need masks. It's really terrible. So the one place where I depart from my colleagues, and I make my colleagues very uncomfortable, I say, yes, work on the message, yes, communication strategy, but unless we make the effort to take down the anti-vaccine content on the internet and go after some of these very aggressive anti-vaccine groups, we're not going to win this battle. And that takes us into a very different place outside the biomedical realm, and that's a larger discussion for a different day. But I think we're going to have to do both, improve our communication strategy and message, but also take down the bad stuff.
Good point. Kind of following up on that question, in working outbreaks and at local levels, it's often you find that the scientists and the responders, the clinicians, are usually the least adept at understanding what's going on at the social level. And because you're so busy doing what you have to do to keep people alive and keep people from being infected, and we have kind of a lens that we don't look through. And I found that anthropologists and sociologists and of course psychiatrists that are available during these outbreaks, they sometimes help to kind of help you watch you. As I was told the other day, you're flying the plane and you're building it at the same time in your outbreak or pandemic situation. So they're kind of sit there to kind of say, hey, you should put that part there. So I think we need to involve other people. And I agree with you that it's the doctorate degrees that have been given for social media is quite large, and the public is using those. And I think we have to message back. The other big point too is tribes. There's this concept called tribes where people listen to their tribe leader, and we as humans, we all have tribes that we listen to. So the message that I might say is different from the message that one of you may say to your group. And so I think we need to be cognizant of that and have consistent messaging across tribes that at least the message gets there. What are some other? Are there social media tools and policy tools that we can use to help do this in your experiences?
I think it's very important to see the influence of the social media on the population, and I think it's a global issue, especially here in our region. I think we can use those people to more educate the patients about because people are now is not listening to the scientific point of view. They are listening more to those influencers on the social media that I think we can use some of them to transfer the information or the instructions that we want to have to our population, especially I'm saying during vaccination. When we have, as Dr. Ziad mentioned, inshallah, in the end of this year, we will have a vaccine. We need to have really some sort of big media coverage for that, and with this we can use the social media. I think it's good to use it.
I think there's a new term now that's out. It's called weaponized health communication, and it refers to what the Russians are doing. I mean, they are flooding social media with negative messages about vaccines and COVID-19 in particular, and it's coming out of the Russian government, including RT, Russia Today, which is their big disinformation empire, and it's causing a lot of damage right now. And so, again, figuring out how to counter all the Russian bots and trolls, which has created this, and this again needs to be taken on at a very high level. That anti-science disinformation is one of the large global destructive forces. I mean, the World Health Organization talked about vaccine hesitancy in 2019. I think in 2020 they're calling it the infodemic, and it's not only in health, but it's particularly strong in COVID-19. I think we need to call the Russians out about this at the G20 summit. This is going to be really important, and it's not only the Russians. I mean, we're getting a lot of misinformation coming out of the Murdoch media empire. News Corps, Fox News. They've been amplifying messages that say, well, the deaths from COVID-19 are really not due to COVID-19, it's due to the comorbidities, fake ideas around herd immunity, saying herd immunity will occur 22% and things like that. So how do you take this on between the Russian government and Murdoch media empire? And this is something that we're not going to do on a Zoom call with a bunch of scientists and physicians, but this is something that needs to be taken on at a future G20 summit at some point. It's because it's almost as damaging as the virus in some ways, in terms of causing people not to take adequate public health measures seriously, preventive measures, and not take vaccines as well.
Good point. Any other comments from the panelists? Moving to another question from the group was, what are the non-medical reasons that are affecting and potentially hindering vaccine development? And I'll add to that, what's the next major challenge in COVID-19 and reaching a normal life again? We've selected the vaccine, we've got distribution plans in place. If everything goes well, what is the other non-medical issues that are going to be facing us as we move forward?
I think the next challenge would be if the virus mutates, and then all the work that's been done over the last 10 months, doing all the work and the research trying to find how to combat this virus will be all gone. And I know I saw a Reuters report this morning that talked about a new virus mutation in the Netherlands, which are infecting minks, and there's a huge culling process happening, where they're killing all the minks in Netherlands because they found the same mutated virus in human. And I don't know the details of the genetic changes and syndrome in the human, but it's something that we would worry about, especially if it's something that the vaccine will not have any impact on.
That's a good point.
Yeah, it could be that we need to develop veterinary vaccines as well to save the mink industry and to look at others. I think that I totally agree. I think that's something that's going to be absolutely essential. But I think, again, the other message is being able to explain that vaccines are not magic, that we are still going to need other public health measures. I think one of the problems with the White House is it tended to veer towards these magical solutions to solving the epidemic. First it was hydroxychloroquine, then it was remdesivir, now it's vaccines, and the way it's often portrayed is like when we developed the Salk polio vaccine. All the journalists were brought to the University of Michigan auditorium, and they pulled back the black curtain, and the vaccine worked, and everybody went dancing in the streets. And it's not going to work that way. These vaccines are going to vary in their level of protective efficacy. Some will likely be partially protective early on. People will still be shedding virus in their upper airways and releasing a lot of virus around. They'll be partially protective at best. So making people understand that vaccines will be important and will be in a much better place next year at this time than we are now. But they're still going to require a lot of situational awareness among the population, staying close to the news and recognizing that the first vaccines will be companion technologies with existing measures, not replacement technologies. And again, none of that is really being effectively messaged.
And Dr. Alabada, what do you see as the business side of this? Where are the problems that might exist in keeping the industry and business involved in this versus relying on the government to continue the support?
Yeah, I think one of big challenge that also we face it in COVID-19, the capability of the companies that in certain countries, we cannot really, we don't have the infrastructure to even manufacture these kind of very successful vaccine to all population, and especially if we have a new vaccine, a new viruses that we have it all, whether it's COVID 2021 or whatever, the capability and the infrastructure are really very poor, especially in our region. That's why it's a big issue for the government, with the help of the private sector, to build this capability for any future, I think, future pandemic or epidemic in our region. I think these kind of issues, it will be a big challenge when we discuss to localize even fill and finish in our country to just cover the Saudi Arabia population. It's also, you need an infrastructure, cannot be done by overnight. It needs at least 2 years to build a facility with high quality, that at least you make sure that the good manufacturing practice that we have, it is really matching the international one. All of these challenge, it should be public and private partnership, because we are in one position that both of us, we have from the investors' point of view and from the government, both of us should work together in order to try to protect ourselves, our families, our countries from this virus, and for any potential virus will come, at least as a healthcare will be prepared for it.
Great, thank you. We just closing out. Does anybody have any pressing thing they would like to share with the audience before we close out for the day?
I think the only thing I would like to share is, from my time as U.S. Science Envoy, and in the book I write, the new book, I identify Saudi Arabia is extremely vulnerable to emerging infectious diseases, and the vulnerabilities even before COVID-19 are due to its location in a pretty volatile part of the world where there's conflict and collapse of health system infrastructure in Iraq and Syria and Yemen, and so that's a vulnerability. I think the high rates of non-communicable illnesses like diabetes, I think that's a vulnerability, and its susceptibility to climate change. You're seeing these unprecedented high temperatures in the region. So all of that means that the kingdom does have that vulnerability to epidemic illness, and I think there needs to be and I think there is this, and I know Ziad's been Dr. Mamish has worked hard on this to raise awareness that this is not just a health issue, it's a security issue for the country. And making that investment in biotechnology infrastructure, in vaccine development is going to be absolutely essential, because right now, for all practical purposes, there's no capability for making vaccines in the region. I think the only country that makes new vaccines is Iran, to some extent. But basically, in North Africa, the Middle East, there isn't really that capability, and Saudi Arabia has the ability to do this if they've got a lot of sophistication. Some of the best universities in the Middle East are in Saudi. There's a lot of biotechnology sophistication. There's the venture capital business sophistication. So all the pieces are in place, and I think this needs to be a priority.
Well, thank you all, and thank you for being on the meeting today, and appreciate your time and your insight. And we'll now move on to the next session. Thanks.
Thank you.
Thank you.
Bye-bye.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound, where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
1, 2, 3, 4. Test 1. Test 2. تست 1 تست بسم الله السلام عليكم بسم الله
لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Our planet never ceases to amaze us. The best is always waiting to be discovered. Every corner hides unseen treasures. In unknown places, treasures of breathtaking beauty that leave you spellbound, where you can reach new heights and feel on top of the world. Run your fingers along the relics of a rich past. Discover new places where you can leave your mark. Be the first to witness a land of fascinating journeys. Where in the world could this be? Saudi. Welcome to Arabia. Get your visa now.
مملكة استضافة قمة الافتراضية لقادة العالم في العاصمة الرياض في لحظة تاريخية غير مسبوقة.
إن علينا جميعا مسؤولية العمل معا والتعاون مع شركائنا في العالم لخلق بيئة يزدهر فيها العالم.
Welcome to this session. It's my great pleasure to welcome you all to this closing session. We've had a great time, and this evening it gives me great pleasure to introduce Professor Adrian Hill. I've known him for a long time. He's the director of the Jenner Institute and Lakshmi Mittal Professor of Vaccinology. He founded the Jenner Institute in Oxford in 2005. And this is an initiative aimed at accelerating public sector vaccine development for a range of infectious diseases. And he partnered with the Pirbright Institute on veterinary vaccine development. The Jenner Institute is now the largest academic vaccine center globally, with clinical stage vaccine programs against 13 diseases. I've worked more closely with Adrian on malaria vaccines, but he's got such a nose for vaccine development that when the COVID pandemic came upon us, it really came as no surprise that Adrian was at the forefront of leading the Oxford vaccine. And so it's a real pleasure today to see what's going on in the clinical trial of the vaccine developed from Oxford. And it's a real pleasure to host Adrian in this webinar today. So over to you, Adrian. We're looking forward to your talk. Thank you.
Thank you very much, Faith, and thank you to all the organizers for inviting me. Can I say, first of all, what a fantastic conference this has been. You have persuaded some fantastic speakers representing so much that's going on in COVID vaccine development to contribute. It's certainly been the most comprehensive meeting I've been to on this subject, going all the way from vaccine design through clinical trials to the great challenges of vaccine biomanufacturing and deployment and global supply. And it's really been great to see an update on that. And also wonderful to see so many contributions from young Saudi scientists, including one of our own experts who trained here with Sarah Gilbert and Eiffel Harvey. So it's great to see all of that, and thank you for asking me to say a few things towards the end. Now, can you see my slides, Faith, or do I need to switch?
I can see your slides, Adrian. I think we're all right.
So this title, Some Learnings from COVID-19 for Vaccines, is a very personal, very general, high-level overview. It's not learnings from COVID-19 vaccines, it's learning from the whole experience for what we could do about developing vaccines better in the future, maybe less expensively, certainly more quickly, and so on. But it's very important to understand that a lot of what I've said has been informed by the experience of being part of a team, not the leader of a team, part of this team of 6 principal investigators from the institute here in Oxford, the Jenner Institute, who came together very rapidly at the end of January this year with a range of talents. So, for example, Andrew Pollard, our chief investigator, the only one of us who has led a phase 3 trial before, and of course an advisor to the UK government on vaccines. He's been horrific, doing not one trial, but several trials around the world, as I'll come to. Sandy Douglas, a younger investigator, leading on improving the manufacturing process for vaccine scale-up, not something you usually find in a university, that sort of work. And then, of course, 2 senior immunologists, Tess Lamb and E.T. Ewer, doing an immense amount of immunomonitoring in the trial. And most importantly of all, Sarah Gilbert in the bottom corner here with myself, who designed the vaccine, had done lots of the background work on emerging pathogens and chimp adenovirus vectors. Now, I'm not going to talk about that program. You've heard from Sarah already in this conference. And believe you me, she doesn't go to a lot of conferences online these days. She's just too busy, so very glad she made this one. So I've put together a few thoughts on things that have emerged from being part of this response team, if you like, that might be interesting or helpful for doing things like this in the future. And at the end, I'll identify some problems that I think we as a global community on vaccines really need to think more about, and we'll probably never have a better time to access support from government and elsewhere for righting these deficiencies. So this slide you know well. It's worth rehearsing because the numbers really are extraordinary if you stand back and look at what's been going on this year. 47 million clinical cases reported. Of course, there are many more that are asymptomatic. The infections, maybe well over 100 million infections, over 1 million deaths, tragically, attributed to COVID-19. But then in the vaccine field, 47 vaccine candidates in the clinic, not in somebody's lab being worked on. And of those 47, 10 of them are in large-scale phase 3 trials, with at least 4 hoping to report efficacy results by the end of the year. That is not only has that not happened before, this is unprecedented speed tackling a pathogen that we didn't know existed a year ago. So can we learn anything from that? And is this a guide as to how we could do vaccine development faster? Faith and I have been involved in malaria vaccines for a long time. Certainly I have been for nearly 3 decades now. And frankly, at times it's been frustrating that things take so long. It's not that the field is short of ideas, it's short of money to assess those ideas and take them into the clinic and evaluate whether they actually work. So my background here is that I am at the Jenner Institute, which, as Faith told you, we started back in 2005, really to bring together people in Oxford who were running their own show, but not accessing all the capabilities that you need to move vaccine candidates from design into clinical development and beyond very rapidly. Picture you will recognize the famous photograph of Edward Jenner vaccinating his gardener's son back in 1796. But the 3 pillars of the Institute that we identified on day 1 were global health vaccines that would make a difference. I think COVID-19 certainly comes into that list now. Translational research, trying as much as we could to test things in humans as well as in small animals, and the score now is 55 vaccine candidates made for clinical trials. And of course, One Health as well, leveraging the know-how in the veterinary vaccine field, and ideally their cost base, which is much less expensive than human vaccine development. So it's become clear even to politicians who don't pay very much attention to science in normal times that vaccines are a good thing for pandemics. There isn't too much controversy about that, even though there are lots of people and anti-vaxxers who think vaccines are just a bad thing, full stop. But it's equally true, I would argue, that pandemics are good for vaccines and for the vaccine field. And frankly, we have an opportunity here to try and get things more aligned, not just to produce better vaccines and to do them for more diseases, but to get an infrastructure in place that doesn't take us so much by surprise next time, and where we don't have to do lots of things that have been irritating in this pandemic, like explain to endless newspaper men that having a pause in your clinical trial doesn't mean it's a disaster. It's something that happens in very large-scale clinical trials all the time. But the most exciting thing for me about this whole year has been that we are undertaking what you could call the grandest global experiment in vaccine technology assessment ever undertaken. Now, that's not the primary objective to figure out which is the best technology, but that is really what is happening. So, you know, if you fantasize about how you would evaluate vaccine technologies, you'd come pretty close to saying, ideally, you would take thousands of global vaccine researchers all around the world doing totally different technologies, incentivize or invite them all to work on just one vaccine target, ideally one antigen from that pathogen, which is largely what's happening, to use their favorite vaccine technology, which we tend to do anyway, to work at record speed with a starting gun fired in, say, the middle of January 2020, and to go and evaluate the safety, immunogenicity, and efficacy of their candidate vaccines, not just in animals, but also in humans in clinical trials, of course, with GMP manufactured product for those trials. And with, for the first time, certainly in my career, pretty unlimited funding available for a rapid response, certainly for the most promising candidates. And then for an encore, they'd be invited to make billions of doses of their vaccine to be available within a year. Now, if I'd told anybody that 12 months ago, they'd have certainly stopped buying me drinks when I was making up that fantasy. But that is exactly what has been happening in the last 12 months. And the data are being produced and published at record speed, and we are learning a lot. And I'm not going to go into the technicalities of the benefits and limitations of different vaccine technologies. The data will be there for us to compare. So if you really want to know about, you know, what vaccine is good for that type of immune response with the same antigen compared with different platform technologies, this is possibly a unique experience, because I suspect for the next pandemic, we won't be assessing 200 candidate vaccines. People will know which ones to go with for which application. So, in summary, what do I think we have learned? And this is a very personal view. Well, firstly, and I feel pretty strongly about this, normal or peacetime vaccine development is far too slow. And if I made that point, and I did many times in the past, people would come back to me and say, oh, well, you know, safety is so important. You have to do things safely, which, of course, I knew a year ago was a nonsense, because the reason it's slow is nothing to do, well, yes, we do trials safely, but the reason that normal vaccine development takes a decade, and this can be done in a year, you're doing the same sorts of trials with the same review bodies, the same hurdles to overcome. It's all to do with the time it takes to do a lot of the administrative stuff that we have to do. Yes, that's leading to safety considerations, but the field has shown you can get lots of vaccines through in a year, right to phase 3 and efficacy assessments. Now that needs a lot of people to align. It needs regulators to prioritize particular vaccines. I don't think we'll generally be making vaccines in a year in the future, but you know, 3 years would be a bit more realistic and sensible than 10 years, which is the average these days. Then some of you on the call will probably agree with me that vaccines and vaccinologists are undervalued. We did shout after Ebola. We made a fuss with avian flu and with swine flu. And we pointed out that the risks were there of something much nastier than swine flu, which wasn't very nasty at all, coming about. And ecologists and others and virologists produced data on what the risks were, and epidemiologists predicted what might happen. And well, it's happened. There's a major global pandemic. Luckily, not with the most lethal of the pathogens that might have appeared, but we should have been better prepared, and some money was allocated. But when you remember that the IMF last month calculated that the likely economic cost of COVID in terms of lost economic development is of the order of tens of trillions of dollars, you know, a bit more investment in putting out this pandemic through vaccines or being better prepared would have been good value for money. Third point is that major vaccine centers do need suitable strategic funds. We all write grant applications as academics. If you're in a company, you spend your time raising money to do more vaccine work. If you're a vaccine company, that investment will be a bargain. If even a single one of the vaccines that are currently in development happens to work and happens to control the pandemic and bring everything back to normal, even if it takes another year, it will still have been a fantastic investment of time. So vaccinology is probably, as you know, one of the most diverse sciences, going all the way from structural biology and immunogenicity design to health economics and logistics of deployment of vaccines and so on. And the specialties you need in between are very, very numerous. Nobody can do it all themselves. You need critical mass, and I would argue you need to be in a major multidisciplinary area where you can access that expertise. There are some fantastic vaccine technologies available. It was wonderful to see RNA vaccines coming through in the last 6 months with lots of data in large numbers of people, showing that a lot of what some people thought was hype, it was real, and they are quite immunogenic, certainly for antibodies, and can be manufactured very quickly and look safe. But on the other hand, investment in manufacturing capacity has been, I'm not talking about RNA vaccines now, I'm talking about vaccines in general. We will hopefully in a few months have some very good vaccines, but we won't have billions of doses of those. And that's not just because that costs a lot of money to manufacture. It's literally because we haven't got places to manufacture them. If you want to make 8 billion doses to cover the world, or 16 billion, because there are 2 doses per vaccine, there is just a lack of manufacturing capacity. And then my final bugbear, and I, have interacted with regulators since the late 1990s. I've never had the problem of having access to lots of regulators who have different views on what should be done and what can be done quickly. So let me try and illustrate that with just one slide on our local vaccine program. This is the ChAdOx1 program that Sarah Gilbert and others have been working extremely hard on. This is now about 55,000 subjects being enrolled or enrolled in clinical trials. In the UK, in Brazil, in South Africa, in the USA, in India, and more recently in Kenya, Japan, and Russia, trials are also underway. And of course, this has been partnered since late April with AstraZeneca. The difficulty in a sentence is that every one of those regulators, of course, is sovereign. And they may well have different ideas on what they require in a clinical trial. And essentially, this is one clinical trial program. We're not going to have loads of efficacy readouts. We're going to have a combined readout from multiple countries. And when you talk to different regulators, it is quite interesting how different a view they can take on how many subjects you need to have for a safety database, what level of efficacy is needed to license the vaccine, what level is needed for emergency use approvals. And there isn't actually an organization out there whose job it is to bring the regulators together. Now, big pharma people know this all the time. They do late-stage development, and they talk to regulators. And I often used to wonder why vaccines and drugs got licensed at different times in different territories. I now know it's because different places want different, have different requirements. So that is really bad for a pandemic. If it's for an ordinary vaccine, which you can wait a year to go somewhere else and get licensed, that's much easier. But, that list of countries is obviously incomplete in the sense that you want a COVID vaccine to be global. And it's not just we are facing this problem. Anybody who wants to make a vaccine for global supply has to somehow address all these somewhat different regulatory challenges. That speed is important, I don't have to suggest that, but some technologies that have been badged as rapid are not always the fastest. I'm pretty reliably informed by people who were in China at the time that it was actually a draw in terms of the race to get into the clinic, that an RNA vaccine, Moderna's in the US, and CanSino's vaccine, actually started dosing in their phase 1 trials in the middle of March on the same day. And not everyone's regarded viral vectors as a rapid technology, but they certainly moved quickly. Capacity is needed all along the pipeline, from design to immunogenicity in animals to comparing your safety profile with what you would expect in animal models to looking at lack of disease enhancement. You will remember back in March, this was the hottest issue. Were vaccines going to do harm? Was there going to be immunopathology like there was in some SARS-1 animal models? Well, so far, touch wood, nothing bad has happened in any vaccine trial in terms of disease enhancement. You need GMP, you need access to it, doing trials quickly, obviously, and then scale up of manufacture. Lots and lots of problems. Just to pick one, GMP facilities are not standing there idle with a staff highly trained and in place, ready to go, awaiting an outbreak. And that consideration on its own has stopped some GMP facilities being built to deal with outbreak response. And as I'll highlight it in a few minutes, immunomonitoring can be rate limiting. Lots and lots of us do lots of immunomonitoring. That's slightly different from having the validated assays or at least qualified assays in place that you can use to present to regulators within weeks or months and haven't time to perfect.
Test 1, 2, 3.
So what do you need to enable a rapid response? This is just some examples from our experience. It's been helpful to have a suitable platform technology. The CHAD vector that we've been using was in the clinic many times before. It's been in 6,000 people before, or the CHAD vectors as a whole have been in 6,000 people before COVID, in many tens of thousands by now. And it's a very versatile technology. You do need a bit of practice at doing these. We have a viral vector core facility that's made many hundreds of these viral vectors. So if you wanted to work first time, it's not just good enough to have the recipe and the method. You really need to have relevant experience and trained people. Regulators really like it if you have evidence that the platform is safe. And that's why we were able to start our phase 1 trial at the end of April and a phase 3 trial at the end of May. That was the first COVID vaccine to go into phase 3 because the regulator we got approval from had seen about 10 different CHAD vectors already with many, many people safely dosed. And they were happy to allow us to start vaccinating not just 1,000 people, but thousands of people within a month or two. So that is my next point. Supportive regulators are absolutely crucial. Whisper it quietly, but different regulators have different amounts of knowledge and experience of different vaccine platforms. That's an issue, and it's understandable if a regulator in a given country has never seen an application for, say, an RNA vaccine before and isn't sure what's needed, that is going to slow things down. And having our own GMP manufacturing facility in the university, the Oxford Dental Biomanufacturing Facility, was crucial to getting a small batch made very quickly. And they're good at that because they've made 20 different CHADs before for clinical trial. The other thing that is perhaps less intuitive is that the hottest and most interesting vaccine technologies that were in Science, Nature, and the top journals just a year ago are not always the most useful. And remember, overall investment in science and vaccinology in particular is really pretty highly skewed to things that are new, much favored over things that are old. And remember, of the 4 vaccine types that have been developed, RNA is the newest, but inactivated vaccines and Vector Pasteur are the oldest. It's not obvious that one is better than the other when you compare the immunogenicity results. Of course, investment is far greater in therapies than in prevention, and that's one of the problems in life being a vaccinologist. Things have to cost less and get less investment. Structural vaccinology is very much flavor of the season. If you double immunogenicity by sticking a few prolines into the right part of your favorite antigen and fixing the structure, you'll get a paper in a top journal. if you say double immunogenicity. If you double immunogenicity by using a better adjuvant or just changing your dosing regime, you'll do well to publish it at all. It's a separate paper. The impact is the same. And remember, older technologies, as I just said, can be as good and are often more scalable because people have been manufacturing them for years rather than working out how to manufacture them during a pandemic. Then again, funders, we complain about all the time, not just when they turn our grants down, have a short time horizon. And epidemics have a short time horizon as well. Look at SARS, lots of vaccine development started with all sorts of new technologies, didn't get funded once the outbreak ended. Zika famously went into a large efficacy trial in the Americas with almost 0 cases because people were too late and Zika had disappeared. And vaccinologists need to be health economists. If you don't know whether your vaccine can be useful and justify its existence, so to speak, at a given price, that's a big challenge. And finally, looking locally here, for university vaccinologists, I think this is a great time for underscoring the point that vaccinology is a serious academic subject, a very translational one. Lots of institutions now want to do more translational research. Vaccinology is perfect for that. And a little disappointingly, if you look at those 47 clinical candidates for COVID I mentioned earlier, tabulated by the WHO, only 5 come from universities. 1 in Japan, 1 in China, 1 in Australia, and a couple in the UK. We should be doing better than that, obviously partnering with industry as required, but we should be creating more immunogens that are rapidly translatable. Too many vaccine institutes in universities are too small and too standalone without the full range of expertise that you need, which is crazy because major research universities are generally very multidisciplinary and have experts who are willing to collaborate on translational activities. So we should learn from that. Modern biomanufacturing is flexible. I said we're very unusual in having our own manufacturing facility. Well, if you recognize that biologicals are a huge class that people making monoclonals and all sorts of other interventions or therapeutics need, it should be possible to invest in local biomanufacturing capacity. And critical mass, as we've learned since the JENA started in 2005 and has grown and grown, is usually have the expertise and experience in-house. A couple of things to emphasize that I think you'll all agree are pretty important. Global supply is a huge challenge. Many of you know that we're starting at a bad place. Vaccine manufacturing is super expensive, very highly regulated. There aren't idling facilities standing by waiting for us to book a slot when a pandemic comes along. Most vaccine doses are made in low-income countries, maybe 70% in one, which is India. Most revenue comes from new vaccines in high-income countries. 80% of the market is in wealthy countries. And generally, high-income countries don't license and have sold vaccines from low-income manufacturers. There used to be a good reason for that. I'm not sure there is now. And absolutely no vaccine, I understand, and I've looked quite hard, has been produced and supplied at 500 million doses in a year. And there are lots of companies out there saying we'll produce 1 billion doses next year of our COVID vaccine. AstraZeneca have contracts for 3 billion doses of the shared vaccine, if that works. So, you know, we're trying to do something that not only hasn't been done before, we haven't been very close to doing it before. But there are exciting new strategies that have been discussed, building new facilities, but nobody's really cracked what they should be doing when there's no pandemic, which could be quite important. And some are being built, one only a few miles from here, but those are government funded, and those governments are going to prioritize their particular countries. And some regulators are much more experienced than others, and if you're going to go global in supply, you're going to encounter all those regulators eventually in particular countries. So delays in approval differ a lot today between countries, and some countries even disallow manufacturing manufacture of some vaccine types that has to be. So being positive to finish with, and I am coming to the end now, there are several solvable problems that we just need to work on, really. One is standardization of immunoassays. The same antigen is being used in nearly all the COVID vaccines. We should be able to compare them for immunogenicity. The problem is there isn't even a standard, let alone a global standard, that we can compare to. So we proudly publish results like this. This is from our Lancet Phase 1 paper, showing that we get pretty good antibody responses, as we say, and I think they probably are good, because if we compare them to convalescent plasma, they're rather similar, particularly after the booster dose, as shown there. But what we can't do is get a direct head-to-head comparison, at least as yet, with anybody else's vaccine. We'd like to do that. We should do that. The world wants to know what the most promising vaccines are, and there isn't even a reference serum out there that is used in all the labs that can calibrate these immune responses. A solvable problem, but it has had impact. I'm not sure, for example, we used the most immunogenic Ebola vaccine in the only efficacy trial in West Africa. Secondly, how safe does your vaccine have to be? Do we need 40 people who've been safely dosed, 400, 4,000, or 40,000? They're not random numbers. There is a large country that started using a vaccine, not licensing it, but using it fairly widely, with around about 40 people vaccinated. 400, reasonable clinical trial. 4,000 would be a lot for licensing many vaccines. That would be sufficient. 40,000 is the number that the Americans are looking at for some of the vaccines supported by warp speed. Now, why would you do a larger number than you needed to if the world was desperate for a vaccine? We wouldn't. These are driven by regulatory requirements and a different view on how much risk-benefit ratio difference you would accept. And of course, if the lethality of SARS-CoV-2 was greater than it actually is, you might well accept 40. I think most people wouldn't accept that today. But do we really want to wait for 40,000 while people are dying of COVID? I'm not sure we should. But that discussion needs to be had, and we need agreement. And then the same point really applies to my third example here, sample sizes for efficacy, which are even more controversial. Famously, in one of the early polio trials, 9 versus 1 was enough to license, 9 in the controls, 1 in the vaccinees. In our initial UK plan, we were looking at 5 to 10,000. That's increased, as you have seen on the earlier slide. And warp speed in the US have been looking for around about a 30,000 mark. But clearly, more is better safety database. But if you don't license, people will die for lack of a vaccine while we wait. So we need a sort of consensus approach on that. And then really on my last slide, there are what you might call cultural, but they're really geographic differences in those views. So we know that in some countries, China, Russia, Bahrain, without efficacy data, vaccines are being used in subsets of the population. Some people think that's wrong. I don't. I think it's a valid decision to make. And it should be an informed decision that is based on quantitative information on safety. Other people think it's crazy. I think we need to have that discussion. And again, it helps if you're developing a vaccine to know where we'd want to do this. I've mentioned the US requirement for large numbers. 150 cases in the efficacy trial are being requested by the FDA for licensure of a COVID vaccine. That is high. It's certainly higher than in some other countries, like in Europe. Are both right? Is it just a different approach to safety? Or is it basing what we do in COVID on what we've done before? And, you know, if you're going to go for 150 cases, what if the efficacy of your vaccine is 90%? After 25 cases, you've got statistical significance. Isn't that something the world would want to know about rather than waiting for another 125? And perhaps the way to capture this is to think about what we would do differently if the case fatality rate of the outbreak pathogen causing the epidemic was not under 1%, but was 50%. And it was just over 50% in Ebola. So I think we need different rules and different guidance and ideally agreement between different countries on what you would do according to a risk profile of the infection. Thank you very much for your attention. I will leave it there and be happy to put things in the discussion.
That's great, Adrian. That really left lots and lots of food for thought. Yeah, I really appreciated the sentiments on teamwork. It's really a huge undertaking, and the need for multidisciplinary expertise, I think, can't be overstated. I think it's given us an incredible opportunity to engage with government and to increase our value factor. And I think it's really got the whole world speaking about immunology and vaccines and vaccine development. So we've catapulted on science engagement like never before. And, you know, every news program, there's an immunologist discussing the pandemic or a vaccine. So I think we have got a big opportunity, really. On the other hand, you really laid out the complications and the challenges of trying to go global with vaccine development. And this is new. When you think of the other vaccines are given in childhood, they're given to specific groups. We've never gone global, needed a vaccine for everybody at the same time. And so there's a minefield there. But one of the first things I want to ask, Adrian, you know, when you think about the facilities and maintaining large facilities for global supply, I don't think they're answers, but one thing that perhaps you could touch on was the valley of death in manufacturing vaccines. You know, to me, this is something that's got to be faced. That yes, we could go up with 47, but we know that not everything's going to work. And how you have that discussion with the pressure to solve a problem immediately. I think that's it would be good to have your thoughts on that.
Yeah, key question. I think I would divide it into normal time and into pandemic response. So in normal times, I think what the world really needs is more comparative immunoassays. So when people develop a new vaccine technology, and people are developing those all the time, they tend to test that vaccine technology, read out the results, optimize it so you get protection in a small animal model, and go into the clinic. What's ridiculously hard for them to do is to compare the immunogenicity and indeed efficacy of their candidate vaccine to 5 or 6 other vaccine technologies in mice, just to get a handle on whether it's good, or non-human primates where it's more expensive, but it's getting hold of the other immune. And you could even avoid that on immunogenicity if you had a reference panel of sera that you could compare with, say, 5. We don't know what the leading vaccine technologies are. There's been a shakeout. You know, you either have viral vectors, or you have RNA or DNA, or you take a inactivated virus, which goes back a long way, but that doesn't mean that it's bad, or you have some other vaccine approach. And the traditional protein and adjuvant one, I haven't really said anything about. That may be the best technology. It just takes a bit longer to get into the clinic and wasn't highlighted by the COVID response. So, yeah, I think if we can become more quantitative in terms of measuring what immunogenicity you're getting, as well as efficacy in an animal model, which is never perfect for extrapolating to humans, I I think you take out a lot of the casualties of the valley of death. And in pandemics, of course, you don't have that time to do that. So in pandemics, you've got to have standing by platform technologies, and hopefully we won't have 47 or 200 or whatever in a couple of years' time. People will know from learnings from COVID-19 what the top 5 or top 10 are, and nothing's going to be perfect. Some things are going to be more immunogenic for antibodies, others for T cells, some more manufacturable, some more stable at 4 degrees for years rather than, others. So you could then pick and choose what you think is most appropriate for that indication, if you like. And this is something that CEPI and other organizations are doing already. And you can even read their calls, and their specifications are very suitable for pandemic response vaccines.
And Adrian, perhaps to be I failed to find the right word, but let's say, what would you have as acceptable efficacy in a pandemic? I know that's a debate that also needs to be had, but what level of efficacy would we feel comfortable with in a pandemic?
Yeah, well, as you know, the more the better, obviously. But what's intrigued me is that many of the people who know the deployability and the public health setting of these application of these vaccines are coming in with the lower figures rather than the higher figures. So, Sarah Gilbert was shooting for 80% efficacy when the vaccine was going into the clinic. I don't think any of us have changed our minds. We think that's possible. But, that would be great. Very few vaccines are 99% effective. But Department of Health people here are talking about how useful a 40 to 50% effective vaccine would be. Let me just explain their thinking. Of course, they want a better vaccine than that. But if you had, say, a 40% effective vaccine against what you measure, which just has to be clinical disease, it's not protection against severe disease, like most vaccines, it's probably going to be better against death and against severe pneumonia. So it might be 50 or 60% against death or getting into intensive care. If you had that product in the coming months in the UK, that would be fantastically useful to our hospitals and the NHS. If you could just halve the number of people who needed intensive care and ventilation, that would be a huge bonus. So, anything over 40%, given that today we have nothing, would be a good start. 90% is going to be a big race for it, and the manufacturing thing is going to come center stage.
Yeah, thanks, Adrian. So a couple of questions from the audience now, Adrian. There's something circulating, an alert circulating in the media about getting your flu shot, and whether that's going to help you against COVID. Could you speak to that?
I could if I knew anything about it. I think there's very little information on flu and COVID interacting. What's certainly been shown that I've seen is that there's a bit less flu around for this stage of the year in the UK. And if you think about it, that's not very surprising. We're in lockdown again.
Yep.
Masks will work against flu as well as other, as well as coronaviruses. So by controlling coronavirus, we might have a milder flu season. Whether you'll get some other interaction whereby sort of local innate immunity to one will protect you against the other, I'm not terribly confident that that's the case. And of course, flu will make you more susceptible to bacterial pneumonia and so on. So I don't think we know very much, and I certainly wouldn't rely on it.
Okay, thanks. There's another question about reinfection, and I guess this goes to the duration of efficacy of whatever vaccines we generate. So there's now kind of increased panic that there's solid reports of reinfection.
Yeah.
So what does that Yeah, how does that bode for vaccines?
Okay, let me answer that because it comes up all the time. People are worried that because natural COVID infection, when you recover, you may be susceptible to getting a second case. And there are second cases reported, reinfections. I think most still asymptomatic. But, you know, the number is of the order of 5 or 10 or something case reports that have been seen that I'm aware of. And there have been tens of millions of COVID disease episodes, let alone COVID infections. So it may be that this is very small print and very rare, but it is over a period of a month to less than a year. So that kind of hints that, you know, it probably won't last 3 years, the protection you get from natural infection, which would not be unusual. I think what I really want to emphasize is that if you have a pathogen that gives you short-lived immunity, it doesn't mean you can't vaccinate against it. And when I say that, a lot of people sort of shake their heads and think, that's unlikely. When I tell them that one vaccine can be really good and another vaccine can be really bad in producing immunity, they believe that immediately, and most people would. So that the point is the same. The quality and magnitude of the immune response you get depends enormously on how you generate it. So if you have an adjuvant in with a protein, it's a very different mechanism putting a virus into a cell where it's expressing the virus, the antigen intracellularly and so on, and the result is strikingly different. Well, if it's a coronavirus that manages to evade the immune response and not generate much, that's a clever coronavirus. It will do well. It will infect a lot of people. But that mechanism that coronavirus has doesn't mean you take a single antigen from that and put it into an RNA vaccine or an inactivated protein or whatever. It will underperform. And we know this in malaria. We know this in, malaria immunity is short-lived. You can make vaccines that will be very highly effective for a year, but not for 10 years probably. So they are separate things. So I think that's good news. You can do better than nature with a lot of current vaccines.
Great. Perhaps a favorite question for you. What's the most trusted parameter about T-cell immune responses?
It depends on what you're asking the T cells to do. And the answer will be to a degree specific to the pathogen you're trying to protect against. For many viruses, an ability to lyse virally infected cells and kill the virus at the same time is extremely important. They tend to be CD8 T cells rather than CD4 T cells, but some CD4s can do that as well. To me, the two biggest parameters in terms of T cell responses are magnitude, how many T cells per mil of blood, and increasingly, and this is quite new information, is location. So you can have fantastic circulating CD8 T cells that will do nothing against a lung infection if they don't come into the lung. And likewise, in malaria, we need our T cells, as you know, in the liver. So, T cell inducing vaccines are still in their infancy compared to antibody inducing vaccines. But for many of these microbes, I'd much prefer to use a vaccine technology that gives you good antibodies, but also gives you measurable numbers of T cells.
Great. Another question on efficacy, Adrian. How would we account for genetics and environmental heterogeneity whilst administering COVID vaccines? I guess the question is really looking at how genetics and environment might affect efficacy.
Is it the genetics of the host now, not the virus?
I think this is the host.
Yeah, no, it's absolutely fascinating. So one of the other extraordinary things that's happened this year is that genome-wide association studies have been done on very large numbers of patients with COVID, bigger than in very common diseases like tuberculosis, for example. And there are some quite striking findings. ABO blood group was one of the first loci to come up. More recently, there's been a much more clear-cut result for a gene on chromosome 3, on the short arm, looking at or where you find a cluster of chemokine and receptor-related genes. It's not entirely clear which of those genes it is, but it's a very big effect size. And what's intriguing is that when you look at the global distribution of the susceptibility gene, or susceptibility allele strictly, you find it's reasonably common in Europeans. It's very common in South Asia. It's very common in South America, and it's absent in Africa. So maybe some of the control of COVID in Africa has not just been African governments doing really well in screening early and looking carefully, which they have done in many places, I'm told, but the fact that Africans are a bit more resistant naturally, genetically, to this virus than other population groups. And I'm told by people in this country, in England, but that's not what we're seeing. You know, in England, in London, if you're an ethnic minority in the UK and not white, you're more susceptible. That's almost certainly socioeconomic and not genetic.
That's thanks, Adrian. Just while you mention Africa, there's one question about what's the best strategy for securing vaccine distribution equitably in low-income areas, based on our experience with malaria.
Well, we had a great talk from CEPI and COVAX earlier today at this meeting, which I think is super impressive. Lots of vaccine going into that fund, coordinated by Gavi, and there to be distributed on an equitable basis. So I think that's a really good start. What we did locally when we were out licensing our university vaccine was we deliberately, and we turned down certain companies because they wouldn't agree to this. We insisted that whatever company took this on did two things. Firstly, they aimed for global supply, not by manufacturing it all themselves, but finding partners like the Serum Institute of India, a huge vaccine manufacturer, who they would sub-license to or whatever, and that's worked out really well. The Serum Institute have made a lot of the Chadox vaccine very recently. And the second thing was we insisted that the price, at least during the pandemic, would be as close to cost of goods as possible. So if you look at what the US government bought or contracted to buy the Chadox vaccine for, it's around about $3 a dose, which is a lot less than most other vaccines. And that's for a country that's not particularly poor. So I think, you know, there's a duty on vaccine developers during a pandemic response to try and have public health impact as globally as possible. But we also need the structures in place, like COVAX, which has been set up by CEPI and by Gates and by Gavi and others, and hopefully it will persist to make sure that there is a mechanism to try and do this, at least.
Fantastic. I'm told we're out of time, Adrian. Thank you so much. That's been a really thought-provoking session, and it's just great to hear how, to just get your thoughts on it as a leading vaccinologist. It strikes me that the way that we have so many advances in technology and all the omics and everything, we need some on the administratomics and regulatomics and distributomics. There's quite a bit that needs to still catch up. But I just want to thank you very much for really enlightening webinar, and I'm happy to close this session. Thanks a lot. Thank you.
On behalf of the organizer, Ministry of National Guard Health Affairs, G20, Saudi Secretariat, King Abdullah International Medical Research Center, and King Saud ibn Abdulaziz University for Health Sciences, we will thank you for attending this important forum that has detailed all the plans in regard of COVID-19 vaccines. In the last 2 days, we have learned a lot about vaccines, their readiness and distribution plans. We thank all our partners and sponsors who made this forum possible that include University of Oxford, Kellogg College, Saudi Health Council, CP, International Industrial Development, National Industrial Development and Logistics Program, Nature Research, MRI Global, Deep Knowledge Group, Bring Hope Humanitarian Foundation, CFI Middle East and Africa, and Zoom. We would like to present you with few recommendations out of this forum that would be read by Dr. Mohammed Boujalal, Chairman of the forum.
It has been a real pleasure to chair this important forum and to hear to all the best talks. These are few points that we have taken as recommendations and as point that we have concluded from this forum. The first, diversity of the type of vaccines is good to increase the ability to protect different population and races with different genetic background who might develop variable immunity or sensitivity to one vaccine compared to others. Second, each vaccine may create or enhance a strong immunity for different durability that could last for months or years and may need a booster vaccine, but even a short immunity time might be efficient enough to break the chain of transmission. Third, further dissections of the immune system response to COVID-19, SARS-CoV-2, and all viruses to inform the discovery of new immunogenic or therapeutic targets for future vaccines and drug discovery and development. Fourth, time to consider combination therapy that include vaccine, adjuvant, and antibody, together with small molecules to enhance the vaccinations. Fifth, keep safety measures such as social distancing and mask is in place for now. These measures are providing to be efficacious in lowering the disease transmission while we wait for the therapy and the vaccines to be developed and marketed. 6. Increase the investment in basic research with a focus in infection disease that include vaccine and virologists to avoid similar Earth lockdown as COVID-19. The basic research that was performed for MERS and SARS has enabled the rapid development of the vaccines candidate for the COVID-19. 7. Better alignment of regulator and vaccine developers would speed up the vaccine approval. 8. Increase and maintain transparency in the vaccine development strategy and share data to enhance the public confidence on vaccines. 9. Increase industry academia collaborative effort during the pandemic to increase our chance to develop faster, efficacious, and safe vaccines and therapies. 10. Increase educational programs for vaccination science to avoid the resistance of population to be vaccinated and enhance confidence of population on science and science-based therapies. Increase the scientists' public engagement and talk to each other to build the trust. 11. In time of pandemic and other devastating disease, we must stop labeling or calling vaccines therapy based on country of origin. Humanity is looking for efficacious and safe vaccine, regardless of its country or race of origin. 12. Increase measure of fair vaccine accessibility and affordability to be able to vaccinate vulnerable population in countries with low GDP. 13. Work with the local leader to gain trust in the vaccine so it will be distributed to their communities. 14. Prioritizing the investment in innovative medical research technology in general and in vaccine development, production, and distribution in particular, and not just for the vital importance to protect lives, but also protect the public and global livelihood, including social life, economy, and mental health. 15. Enhance manufacturing and cold chain supply capability in the developing and low-income countries. 16, and last, continue to host annual global forums for top vaccine scientists from around the world to engage, to encourage goodwill, transparency, and collaboration. And thank you very much for attending this forum. Thank you. Thank you, President.
Thank you, Dr. Mohammed. Now we would like to ask the members of the organizing committee to give thanks words. I will start with Dr. Marwan Baker. Dr. Marwan, can you hear me? Dr. Marwan. Okay, Dr. Jean. Dr. Jean, can you hear me? Julia?
Yes, we.
Yeah, please, Dr. Ahmed.
Okay. Thank you very much. Actually, it's an echo from my side. Sorry for that. Let me start to thank King Abdullah International Medical Research Center for hosting this virtual symposium. Special thanks to the Minister of National Guard, His Royal Highness Prince Abdullah bin Bandar bin Abdul Aziz. Thanks to Dr. Al-Knawi, Dr. Al-Askar. Special thanks for all the speakers who shared all of this invaluable knowledge with us. I have to especially thank them for putting their energy and efforts to serve humanity by struggling finding a vaccine to fight this pandemic virus. I would like to thank all the 10,000 attendees worldwide, and I hope that it has been informative for them, and really hope that they can advocate it and share it with their colleagues and friends. Thanks for all the sponsors, and let me especially thanks for all my colleagues in the organization committee that you have worked very hard the last 2 months to make this happen. I haven't seen any of you face to face or physically. I hope that we can do it in the near future. But I'm so proud of you. I'm proud of this symposium, bringing all of these people around the world, regardless of the geographical, and fighting for one thing, serving humanity. Thank you very much. Please stay safe.
Thank you, Dr. Baker.
Thank you to the organizing committee. You guys really have done a lot. I have been unfortunately unable to join sometimes, but every time somebody picked up the effort and moved it forward, and it's been a great opportunity to see this team working together. And of course, our presenters, the information they've provided us gives us lots of insight and helps us understand more about what's occurring. I know it seems like there's been ages since this pandemic started, but my reminder to the audience and also to our group is that the pandemic is just starting really in the sense of our response and recovery phase. And much of the story is left, still hasn't been told, and we have to wait for it to unfold in front of us. And there'll likely be a lot of, variation in that depending on where we live in the world. And let's try to make that not be a variation. Let's try to reduce that variation between countries. I look forward to the future with caution always, because will we learn from this and be better prepared in the future, but also with a great optimism, because the the promise of science and technology has really been demonstrated here, and it continues to grow, and we're going to be able to do a lot more in the future. And open discussions like this are part of that process. So thank you.
Thank you, Jean. Julia?
I'm muted. I would like to thank the hosts, the organizing committee, and the speakers for participation in this wonderful international conference. I would also like to thank the research and medical laboratory workers and the support staff that are working around the clock to perform the experiments needed to design, build, and test the vaccines for COVID-19. And then I'd like to thank everyone for attending this conference and learning all about the science behind vaccines, and hopefully to go out and tell other folks that they will be safe to use and we'll be able to end this pandemic and get back to normal. Thank you, everybody.
Thank you, Julia. Ahmed?
I would like to definitely thank everyone who made this event happen. Of course, starting by the organizers, by the speakers, and by all the teamwork that make this happen. Thank you very much. It was really amazing to bring all these bright scientists together and to hear all these amazing talks. And I'm sending my warmest greetings and thanks from Oxford to everyone. And we always, by end of a conference like this, we say we wish you a safe trip back home, but actually this time we'll say we wish you to stay safe at home, and hopefully we will meet at some point. Thank you very much all. Have a wonderful day.
Thank you, Ahmed. Margarita?
No. It's been an absolute pleasure to be involved in this global forum. I'd like to thank all of the scientists from all over the world who spoke in the forum. I'd like to thank all of the attendees who attended and all of my fellow colleagues who helped organize the forum. But most of all, I'd like to thank our hosts in Saudi Arabia, who included people from all over the world and went to great effort to include people from every continent, humanitarians, scientists, and they were just so inclusive. And they were so gracious to all of us. So thank you very much to everybody in Saudi Arabia.
Thank you very much, Margarita. Naif?
Right, thank you so much. Amazing 2-day discussing vaccine and vaccine accessibility by the top vaccinologists in the world, and this special events, which comes in this difficult time of the pandemic. We are very proud of the unprecedented effort that has been made by all these vaccinologists and vaccine professionals all over the world, who made the time for making a vaccine reduced from 10 years or so to under 1 year, which is unprecedented in human history. I would like to thank our speakers who made this event an enjoyable and beneficial to all of us. Thanks also to my fellow members in the organizing committee. Thanks to our organizers and sponsors. But the big thank you also goes to the attendees, the thousands of attendees who attended with us in those 2 days from over 90 countries across the globe. Thank you.
Thank you, Dr. Naif. Thank you for your attendance, and we look forward to another forum. Stay safe.
At one time, and with one goal. A journey on a bullet train in which we race against time towards developing a vaccine to save life and humanity. This journey includes 32 experts and 10 scientific sessions, in which we discuss 6 topics revolving around developing a successful vaccine against the COVID-19 virus. From Riyadh, Kingdom of Saudi Arabia, we welcome experts to whom the world looks with hope, and major companies that will lead the way in distributing vaccines around the world. From Health Affairs of Ministry of National Guard, King Abdullah International Medical Research Centre, KMARC, King Saud bin Abdulaziz University for Health Sciences, and the Saudi Secretariat of the Group of 20, G20. We say to everyone, let us proceed with one ambition and with one goal towards a meaningful life, and let us work together to protect the health of our fellow human beings and our future generations.